Good afternoon and welcome. I would first like to remind everyone to please mute your line when you're not speaking. For media and press, the FDA press contact is Shanapa Tatimbanchanchai. Her email and phone number are currently displayed. My name is Jorge Garcia, and I will be chairing today's meeting. I will now call the April 21st, 2022 meeting of the Oncology Drug Advisory Committee to order. Dr. Shisha Shen is the designated federal officer for this meeting, and she will begin with the introductions.
Good afternoon. My name is Shija Chen. I am the designated federal officer for this meeting. When I call your name, please introduce yourself by stating your name and affiliation. We will first start with ODAC members. Dr. Avani.
Dr. Advani, Stanford.
Dr. Conaway.
Mark Conaway, biostatistician, University of Virginia.
Dr. Cristofanini.
Yeah, Dr. Massimo Cristofanilli, Breast Medical Oncology, Weill Cornell, New York.
Dr. Garcia.
Jorge Garcia, Chief Medical Oncology, University Hospital Simon Cancer Center, Case Western Reserve University, Cleveland, Ohio.
Dr. Liu.
Christopher Liu, GI Medical Oncologist, University of Colorado.
Dr. Madan.
Hi, Ravi Madden, GU Medical Oncologist, National Cancer Institute.
Mr. Mitchell.
I'm David Mitchell. I'm the consumer representative to the ODAC. I'm the founder of Patients for Affordable Drugs, and I'm a patient in ongoing treatment for hematological malignancy, multiple myeloma.
Dr. Nieva.
George Nieva, Section Head Solid Tumors, University of Southern California, Norris Comprehensive Cancer Center.
And Dr. Song. Dr. Song, I think you might be muted. So we'll go back to Dr. Song later. Next are temporarily voting members. Dr. Al.
I'm Jessie Al. I'm founding director of Institute of Quantitative Systems Pharmacology in Carlsbad, California.
Dr. Chen.
Andy Chen, Malignant Hematology, Oregon Health and Science University.
Dr. Coffey.
Yeah, Chris Coffey. I'm a professor of biostatistics at the University of Iowa.
Dr. Deal.
Sue Deal, Hematologic Malignancies, Duke University.
I'm going to go back to the ODAC member. Dr. Song, please unmute yourself, introduce yourself, and state your affiliation, please. You're still muted, Dr. Song. Can you give a shot? Okay, we'll come back later. Okay, continue with temporarily voting members. Dr. Dunleavy.
I am Karen Dunleavy. I'm the Director of Hematology at Lombardi Cancer Center at Georgetown University in Washington, D.C.
Dr. Croft.
Mulder Kraft. I'm an internist and clinical pharmacologist at Thomas Jefferson University in Philadelphia.
Ms. Nadine Baker. I am a CLL patient, and I am the patient representative on this panel. And Dr. Tena Rajasingam.
Tena Rajasingam. I'm a lymphoma hematologist and a health outcomes researcher at the Mayo Clinic in Rochester, Minnesota.
Next is industry representative to the committee, Dr. Chen.
Hi, I'm Jonathan Chang. I'm the industry rep. I'm a medical oncologist, and I'm with Bristol Myers Squibb.
Last are FDA participants, Dr. Pazler.
Richard Pazder, Director, Oncology Center of Excellence, FDA.
Dr. Sierra.
Yeah, hi, Dr. Mark Thierry, Deputy Center Director, Oncology Center of Excellence, FDA.
Dr. Gormley.
Hi, I'm a hematologist and the Division Director of the Division of Hematologic Malignancies 2, FDA.
Dr. Richardson.
Hi, Nicholas Richardson, Clinical Team Leader, Division of Hematologic Malignancies 2, FDA.
Dr. Kasman.
Hi, Yvette Kasserman, Clinical Team Lead, Division of Hematologic Malignancies 2, FDA.
Dr. Weiss.
Hello, I'm Thomas Wise. I'm the Director of the Division of Biometrics 9 at FDA.
And Dr. Booth.
Good afternoon. My name is Brian Booth. I'm the Director of the Division of Cancer Pharmacology 1 in the Office of Clinical Pharmacology at the FDA.
Okay, I'm going to go back to Dr. Song again. Dr. Song, please unmute yourself. Again, introduce your name and say your affiliation. Thank you.
For topics such as those being discussed at this meeting, there are often a variety of opinions, some of which are quite strongly held. Our goal is that this meeting will be a fair and open forum for discussion of these issues and that individuals can express their views without interruption. That's a gentle reminder. Individuals will be allowed to speak into the record only if recognized by the chairperson. We look forward to a productive meeting. In the spirit of the Federal Advisory Committee Act and the Government in the Sunshine Act, we ask that the Advisory Committee members take care that their conversations about the topic at hand take place in the open forum of the meeting. We are aware that members of the media are anxious to speak with the FDA about these proceedings. However, FDA will refrain from discussing the details of this meeting with the media until its conclusion. Also, the committee is reminded to please refrain from discussing the meeting topic during the break. Thank you. Dr. Shisha Chen will now read the conflict of interest statement for the meeting. Dr. Chen.
Thank you, Dr. Garcia. The Food and Drug Administration, FDA, is convening today's meeting of the Oncologic Drugs Advisory Committee under the authority of the Federal Advisory Committee Act, FACA, of 1972. With the exception of the industry representative, all members and temporary voting members of the committee are special government employees, SGEs, or regular federal employees from other agencies, and are subject to federal conflict of interest law and regulations. The following information on the status of this committee's compliance with federal ethics and conflict of interest law covered by, but not limited to, those found at 18 U.S.C. Section 208 is being provided to participants in today's meeting and to the public. FDA has determined that members and temporarily voting members of this committee are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have potential financial conflicts when it is determined that the agency's need for special government employees' services outweighs his or her potential financial conflict of interest, or when the interest of a regular federal employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employee. Related to the discussions of today's meeting, members and temporarily voting members of this committee have been screened for potential financial conflicts of interest of their own, as well as those imputed to them, including those of their spouses or minor children, and for purposes of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts, grants, carve-outs, teaching, speaking, writing, patents and royalties, and primary employment. Today's agenda involves discussion of the appropriate approach for phosphatidylinosyl 3-kinase inhibitors, currently under development in patients with hematologic malignancies, and whether randomized data should be required to support its demonstration of substantial evidence of the effectiveness and that the drug is safe for its intended use in the proposed population. This is a particular meeting during which general issues will be discussed. Based on the agenda for today's meeting and all financial interests reported by the committee members and temporarily voting members, no conflict of interest waivers have been issued in connection with this meeting. To ensure transparency, we encourage all standing members and temporarily voting members to disclose any public interest statements that they have made concerning the topic at issue. With respect to FDA's invited industry representative, we would like to disclose that Dr. Jonathan Chen is participating in this meeting as a non-voting industry representative, acting on behalf of a regulated industry. Dr. Chen's role at this meeting is to represent industry in general and not in particular company. Dr. Chen is employed by Bristol Myers Squibb. We would like to remind members and temporarily voting members that if the discussions involve any other topic not already on the agenda, for which an FDA participant has a personal or imputed financial interest, the participants need to exclude themselves from such involvement, and their exclusion will be noted for the record. FDA encourages all other participants to advise the committee of any financial relationships that they may have regarding the topic that could be affected by the committee's discussion. Thank you.
We will proceed with the FDA introductory comments from Dr. Nicole Gormley. Dr. Gormley?
Thank you. Good afternoon. I'm Nicole Gormley, a hematologist in the Division of Hematologic Malignancies 2 at the FDA. I will provide a brief introduction to the PI3 kinase inhibitors and the reasons for discussing this drug class at an advisory committee meeting. This committee meeting is not a typical ODAC where we would discuss the risk-benefit profile of a specific product, but instead we will discuss the class of PI3 kinase inhibitors as a whole, the unique toxicities they present, and the best development approach for future drugs in this class. I'd like to start by providing a brief overview of the drugs in this class and their mechanisms of action. Overactivation of the PI3 kinase pathway is common in malignancy. Activation of the PI3 kinase pathway can occur via several mechanisms, including mutations of the PIK3CA gene or mutations of downstream effector proteins. Constitutive activation of the PI3 kinase pathway is common in hematologic malignancies. The PI3 kinase family of enzymes is grouped into three classes. Class 1A and 1B PI3 kinases activate or inhibit downstream proteins, which affect cell growth, apoptosis, cell cycle regulation, glucose metabolism, and DNA repair. The PI3 kinase inhibitors are targeted immunomodulatory drugs and inhibit different isoforms. The PI3 kinase inhibitors, which have been developed for hematologic malignancies, are listed here. All of the PI3 kinase inhibitors approved for hematologic malignancies inhibit the delta isoform. While idelalisib and umbralisib are selective delta inhibitors, copanlisib inhibits both delta and alpha isoforms, and duvelisib inhibits both delta and gamma. Umbralisib also inhibits casein kinase CK1 epsilon. Of note, alpelisib is the only other FDA-approved PI3 kinase inhibitor. It is approved for the treatment of PIK3CA mutated advanced or metastatic breast cancer and for the treatment of patients with severe manifestations of PIK3CA-related overgrowth spectrum. It is an alpha inhibitor and is not within the scope of the meeting today. The PI3 kinase inhibitors' distinct mechanisms of action result in a differentiated safety profile, depending on the isoform targeted. The delta and gamma isoforms are preferentially expressed on leukocytes, resulting in infections and immune-mediated toxicities. The infections may occur in part because of treatment-related cytopenias, but also because of the modulation of the immune system by the PI3 kinase inhibitors. Infections include pneumonia, opportunistic infections like PCP and CMV reactivation. With regards to the immune-mediated toxicities, the delta isoform is important for T regulatory cell function. It is thought that the decreased T regulatory cell activity and increased CD8 cytotoxicity damages normal tissue, leading to the immune-mediated toxicities associated with these products. Hepatitis, pneumonitis, colitis, and rash have been observed. Younger patients and those less heavily pretreated with more robust immune systems may be at greater risk for these immune-mediated toxicities. The alpha isoform is ubiquitously expressed and is essential to cellular growth and metabolism and glucose homeostasis. Resultant toxicities from alpha inhibition include hyperglycemia and hypertension. To highlight this further, I have included the common toxicities observed with the approved PI3 kinase inhibitors. Of note, there have been high rates of severe, grade 3 or higher adverse events, high rates of serious adverse events, and significant discontinuations and dose reductions due to adverse events. The AEs observed include high rates of infection and immune-mediated toxicities. Copanlisib, the only inhibitor in hematologic malignancies that also inhibits the alpha isoform, has hyperglycemia and hypertension-related toxicity. This slide lists the labeling and other risk mitigation strategies used to communicate the risks associated with these products. Idelalisib and duvelisib have box warnings and communication REMS. The warnings and precautions for each of the products are listed. The first PI3 kinase inhibitor approved in the U.S. was idelalisib in 2014. What is notable is that other than in CLL, where the initial approvals were based on randomized trials, the initial approvals for other indolent lymphomas, follicular lymphoma, marginal zone lymphoma, were based on single-arm trials and were granted accelerated approval. Also of note, in December 2021, the sponsor for duvelisib, in consultation with the FDA, decided to voluntarily withdraw the FL indication. And in February 2022, the sponsor of idelalisib decided to voluntarily withdraw the FL and SLL indications for that product. And most recently, last week, the sponsor for umbralisib announced that they will withdraw the FL and MZL indications. These withdrawals will be discussed further momentarily. There are two approval pathways available in the U.S., regular approval and accelerated approval. Accelerated approval is available for drugs or biologics that are intended to treat a serious or life-threatening illness. The product should provide a meaningful therapeutic benefit over available therapy, and approval is based on an endpoint reasonably likely to predict clinical benefit or an intermediate endpoint. For products granted accelerated approval, there is often a requirement to conduct post-approval trials to verify the anticipated clinical benefit. I'd like to briefly review the evidentiary criteria for approval. It is important to note that drugs granted accelerated approval or regular approval must meet the same statutory requirements for safety and effectiveness. For safety, there must be sufficient information to determine that the drug is safe for use under the conditions prescribed, recommended, or suggested in labeling. For effectiveness, there must be substantial evidence of effectiveness that allows for the conclusion that the drug will have the effect it purports or is represented to have under the conditions of use prescribed in labeling. This slide outlines the FDA-approved treatments for CLL and indolent non-Hodgkin's lymphoma. These products are often used together as part of combination therapy and may be used for retreatment at relapse. The approved classes include chemoimmunotherapy, CD20 monoclonal antibodies, BTK, BCL2, and EZH2 inhibitors, and CAR-T therapies. There are several central issues that we will discuss further as it relates to the PI3 kinase inhibitors, specifically a potential detriment in overall survival in multiple randomized trials, toxicity and tolerability of the PI3 kinase inhibitors, dosing considerations and inadequate dose optimization of several of the products to date, and the limitations of single-arm trials. There have been several randomized controlled trials evaluating a PI3 kinase inhibitor in combination with immunotherapy or chemoimmunotherapy in patients with CLL or indolent non-Hodgkin's lymphoma that have shown worse overall survival compared to the control arm. Notably, the overall survival information from these trials is early and represents a low number of events. Nevertheless, while the trials show a favorable impact on efficacy endpoints, such as progression-free survival or overall response rate, there have been higher rates of death, and the overall survival results are concerning. It is also important to consider the patient population, those with CLL and indolent non-Hodgkin's lymphoma. These diseases have a long natural history, and progression isn't necessarily an indication for treatment. While these are serious and life-threatening diseases, and there is a need for continued development of products to treat relapsed or refractory disease, there are multiple therapeutics with established efficacy and safety. While the PI3 kinase inhibitors have a unique toxicity profile and several trials have demonstrated concerning overall survival results, some of these findings may be related to poor dose optimization. The optimal dose that maximizes efficacy and minimizes safety may not have been identified. Across their class, there's been limited dose exploration. Many doses were determined using a maximum tolerated dose or MTD approach with limited exploration of lower dose levels. For each of the approved PI3 kinase inhibitors, there are exposure-response relationships for safety, but exposure-response relationships for efficacy have not been consistently observed. High rates of discontinuation, interruption, and modification also suggest the approved doses may be poorly tolerated. There has been voluntary withdrawal of approval of 3 PI3 kinase inhibitor indications to date: idelalisib, duvelisib, and umbralisib. Idelalisib for relapsed refractory FL or SLL was granted accelerated approval in 2014 based on a single-arm trial. At the time, 3 accelerated approval post-marketing requirements were issued to verify the clinical benefit. The first PMR was a dose optimization study for chronic administration. The second PMR required submission of the final report and data showing safety and efficacy from study 0124, a phase 3 2-arm randomized placebo-controlled trial of idelalisib in combination with rituximab in patients with previously treated INHL. And finally, the third PMR required submission of the results from study 0125, a phase 3 randomized placebo-controlled trial of idelalisib in combination with bendamustine and rituximab in patients with previously treated INHL. In 2016, the FDA was notified that 3 randomized controlled trials were terminated due to increased deaths in the idelalisib arm. These terminated trials included the 0124 and 0125 accelerated approval confirmatory trials. The third terminated trial was evaluating idelalisib in combination with bendamustine and rituximab in patients with treatment-naive CLL. Several regulatory actions were taken by the FDA as a result of these findings. A limitation of use was added to the label that idelalisib is not indicated for first-line treatment and is not indicated in combination with bendamustine and rituximab in follicular lymphoma. There were updates to the box warning and warnings and precautions. A new PMR was issued to conduct a trial to establish the safe and effective dose of idelalisib in patients with relapsed refractory FL who have no other therapeutic options. The PMR was to be supported by Study 1580. In February 2022, citing challenges in enrollment to the confirmatory trial and inability to provide evidence to verify the clinical benefit of idelalisib in patients with FL and SLL, the sponsor, in consultation with the FDA, decided to voluntarily withdraw the FL-SLL indication from the U.S. market. The second voluntary withdrawal was for duvelisib. Duvelisib for relapsed refractory FL was granted accelerated approval in 2018 based on a single-arm trial. At that time, one accelerated approval post-marketing requirement was issued to verify the clinical benefit. The planned trial to support this was going to be the Duetto trial, a phase 3 randomized trial of duvelisib plus rituximab compared with rituximab alone or rituximab in combination with CVP. The trial was never initiated due to feasibility issues and a changing treatment landscape. Because of the inability to provide evidence to verify the clinical benefit of duvelisib in patients with FL, the sponsor, in consultation with the FDA, decided to voluntarily withdraw the indication from the U.S. market in December of 2021. On April 15, 2022, the umbralisib and ublituximab applications for the U2 combination regimen were voluntarily withdrawn. This was due to updated overall survival data from the Unity CLL trial, which showed an increasing overall survival imbalance in favor of the control arm. At the same time, the sponsor also announced the voluntary withdrawal of the existing umbralisib indications of relapsed or refractory FL and MZL under accelerated approval. The withdrawn indications were under accelerated approval, in which the approvals were based on single-arm trials. With the PI3 kinase inhibitors, we have seen several instances in which the confirmatory trial with randomized data identified concerning overall survival results and concerning toxicities. It is worth underscoring at this point some of the limitations of single-arm trials. In single-arm trials, the safety findings are challenging to interpret. Without a comparator, it can be challenging to attribute adverse events observed to the drug or to the underlying disease. The efficacy can also be challenging to interpret. The responses observed may not translate into true clinical benefit. Comparisons to historical populations or cross-trial comparisons are fraught with limitations. When evaluating single-arm trials for accelerated approval, there is a requirement that the therapy provide a clinically meaningful advantage over available therapy. Given the different temporal conduct of trials, differences in the patient populations, and other changes to standards of care, the comparative assessment can be challenging. Finally, because of the aforementioned limitations of cross-trial and historical comparisons, to avoid these and other biases, time-to-event endpoints such as progression-free survival and overall survival cannot be accurately assessed or interpreted in single-arm trials. Therefore, in a single-arm trial, it is hard to balance the observed efficacy with toxicity to appreciate the true benefit-risk of the drug in the intended patient population. The inability to assess overall survival in single-arm trials is important because overall survival is an objective measure of clinical benefit and is both a safety and an efficacy endpoint. Overall survival incorporates the impact of toxicity and is useful in assessing both short-term and long-term impacts of therapy. We would like for the committee to please discuss the observed toxicity of the PI3 kinase inhibitor class and whether randomized data are warranted with an assessment of OS to support the evaluation of benefit risk in patients with hematologic malignancies. The voting question is, given the observed toxicities with this class, previous randomized trials with the potential detriment in OS, and a narrow range between effective and toxic doses, should future approvals of PI3 kinase inhibitors be supported by randomized data? Thank you for your attention. Dr. Richardson will discuss these issues in further detail.
Thank you, Dr. Grumley. We will now proceed with the FDA presentation with Dr. Nicholas Richardson. Dr. Richardson.
Good afternoon. I'm Nicholas Richardson, a pediatric hematologist oncologist in the Division of Hematologic Malignancies 2 at the FDA. I will be presenting the FDA's discussion on the PI3K inhibitors in hematologic malignancies. As mentioned by Dr. Gormley, this ODAC meeting is not a typical product-specific ODAC. We are here to discuss the class of PI3K inhibitors as a whole, the unique toxicities they present, and the best drug development approach for future PI3K inhibitors that are developed in patients with hematologic malignancies. To support a class discussion, it will highlight relevant data for each of the approved PI3K inhibitors in hematologic malignancies that are shown on the slide. This will be followed by a class-wide discussion. The central issues we would like to focus on today are multiple randomized trials showing a potential detriment in overall survival, toxicity of the PI3K inhibitor class, dosing considerations and dose optimization, and trial design considerations regarding limitations of single-arm trials. The members of the FDA review team are listed here. My presentation represents their collective input. I would like to start with a brief overview of the timeline for the approved PI3K inhibitors in hematologic malignancies and relevant milestones. The first PI3K inhibitor approved in the U.S. was idelalisib in 2014 for patients with chronic lymphocytic leukemia, follicular lymphoma, and small lymphocytic lymphoma. The FL and SLL indications were granted accelerated approval based on a single-arm trial. Subsequently, in March 2016, the FDA was notified regarding 3 randomized trials with idelalisib showing early signs of worse overall survival. This prompted an FDA safety alert and an update to the idelalisib label with updated safety information and limitations of use. In February of this year, the FL and SLL indications under accelerated approval were voluntarily withdrawn due to the inability to provide evidence to verify clinical benefit for idelalisib in patients with FL or SLL. The second PI3K inhibitor approved was copanlisib in 2017. Copanlisib was granted accelerated approval in patients with relapsed follicular lymphoma based on a single-arm trial. In May of 2021, a supplemental new drug application for copanlisib in patients with indolent non-Hodgkin lymphoma was submitted based on the randomized Kronos 3 trial. The application was subsequently withdrawn in December of 2021 to allow for additional analyses of data from ongoing trials. The third PI3K inhibitor approved was duvelisib in 2018. Duvelisib was approved for patients with CLL or SLL and follicular lymphoma. The FL indication was an accelerated approval based on a single-arm trial. In December 2021, the follicular lymphoma indication was voluntarily withdrawn due to the inability to provide evidence to verify clinical benefit for duvelisib in patients with follicular lymphoma. Lastly, umbralisib was granted accelerated approval for follicular lymphoma and marginal zone lymphoma in February 2021 based on a single-arm trial. A subsequent supplemental new drug application was submitted in May 2021 for patients with CLL and SLL based on the randomized Unity CLL trial. Based on ongoing analyses and concerns with the Unity CLL trial, an FDA safety alert was issued in February 2022 for a possible increased risk of death in those treated with umbralisib. Last week, on April 15th, the application based on the Unity CLL trial was withdrawn from the FDA. In addition, the existing FL and MZL indications for umbralisib currently under accelerated approval are being voluntarily withdrawn from the U.S. market. Now we will transition to discussing the relevant data for the approved PI3K inhibitors. We will start with idelalisib, a PI3K delta inhibitor. The issues we will highlight are decrements in overall survival in several randomized trials, PI3K-associated toxicity, and dosing considerations. Idelalisib was granted regular approval for patients with relapsed CLL in combination with rituximab in patients for whom rituximab alone would be considered appropriate. The approval was based on the 0116 study, a randomized placebo-controlled trial that demonstrated a statistically significant benefit in progression-free survival in those treated with idelalisib plus rituximab with an approximate 13-month improvement in PFS with an adjusted hazard ratio of 0.15. At the time of approval, the overall survival information was early with a total of 19 overall survival events, or 9%, with an estimated OS hazard ratio of 0.37, favoring the idelalisib arm. Idelalisib as monotherapy was also granted accelerated approval for patients with relapsed follicular lymphoma or small lymphocytic lymphoma after at least 2 prior systemic therapies. This was based on an overall response rate of 54% in follicular lymphoma and 58% in small lymphocytic lymphoma with associated durability from a single-arm trial. Based on the trial supporting the initial approval of idelalisib, a notable toxicity profile was observed. To mitigate risk, several measures were included as part of the initial approval. A boxed warning used to highlight adverse reactions so serious in proportion to the potential benefit from the drug that it is essential that it be considered in assessing the risk and benefits of using the drug. The initial approval of idelalisib included a boxed warning for hepatotoxicity, diarrhea or colitis, pneumonitis, and intestinal perforation. Additionally, the toxicities rash, neutropenia, and anaphylaxis were included as warnings and precautions. A risk evaluation and mitigation strategy, or REMS, was included with the initial approval of idelalisib. A REMS is a safety program used to ensure a drug is safe and effective for its intended use and that its benefits outweigh its risk. Along with risk mitigation, a number of post-marketing requirements were issued for idelalisib. There was a post-marketing requirement issued to conduct a trial to optimize the dose of idelalisib in patients with follicular lymphoma or small lymphocytic lymphoma. Additionally, 2 post-marketing requirements were issued to verify the clinical benefit of idelalisib in patients with indolent non-Hodgkin lymphoma based on 2 ongoing randomized trials. A total of 4 additional post-marketing requirements for safety were issued. These included characterization of the risk of pneumonitis and to characterize long-term safety across ongoing trials. In March of 2016, the FDA was notified of 3 randomized trials evaluating idelalisib in combination with immunotherapy or chemoimmunotherapy that were terminated due to increased death and severe toxicity in the idelalisib arms. These trials were the 0123 trial in patients with untreated CLL, the 0124 and 0125 trials in patients with previously treated indolent non-Hodgkin lymphoma. Each of the trials was a randomized, double-blind, placebo-controlled trial. The respective treatment arms are shown in the table. This table shows the interim overall survival results for the 3 randomized trials. In each trial, there were more deaths in the idelalisib arm compared to the control arm. Despite a limited number of overall survival events, the estimated hazard ratio for these trials showed the potential for an increased risk of death and harm to patients. The reasons for death in the 3 randomized trials indicate a higher rate of death due to adverse events in the idelalisib arm. The primary adverse event leading to death were infections, as shown in the table. This graph shows the safety results from the 3 randomized trials. For grade 3 or greater toxicity, serious adverse events and discontinuation, dose reduction, or dose interruption due to an adverse event, the rates were notably higher in the idelalisib arms, as indicated by the blue bars in the graph. Even with treatment modifications due to adverse events, the increased rates of grade 3 or greater toxicity and serious adverse events indicate overall safety concerns with idelalisib in the evaluated populations and uncertainty regarding the idelalisib dosing regimen. The safety results from the 3 randomized trials demonstrate that the PI3K-associated toxicities of grade 3 or greater infection, neutropenia, diarrhea or colitis, increased ALT or AST, rash, and any grade pneumonitis are driving the differences in safety between the treatment arms. Shown in the table, the incidence of any grade pneumonitis or grade 3 or greater PI3K-associated toxicities, except neutropenia, are 2 to 3 times higher compared to the control arm. The data from the 3 idelalisib randomized trials led to an FDA safety alert regarding higher deaths and severe toxicity. A Dear Healthcare Provider letter was also issued. Additional risk mitigation measures were implemented. The boxed warning for idelalisib and the REMS was updated to include the risk of fatal or serious infections. The safety data from the 3 randomized trials was included in labeling, and most importantly, the randomized data informed limitations of use for idelalisib. The limitations of use include the frontline treatment of any patient, and that idelalisib is not indicated or recommended in combination with rituximab or in combination with bendamustine and rituximab in patients with follicular lymphoma. The 3 terminated idelalisib trials included the 0124 and 0125 accelerated approval confirmatory trials for idelalisib in indolent non-Hodgkin lymphoma. Therefore, a new accelerated approval post-marketing requirement was issued. The new post-marketing requirement was to identify a safe and effective dosing regimen in patients with follicular lymphoma who have exhausted known treatment options. Study 1580 was an ongoing study evaluating different dose levels and different regimens of idelalisib in patients with follicular lymphoma. The 1580 study encountered enrollment challenges. Ultimately, because of the inability to provide evidence to verify the clinical benefit of idelalisib in patients with follicular lymphoma and small lymphocytic lymphoma, as required per the accelerated approval regulations, the FL and SLL indications for idelalisib were voluntarily withdrawn in February of this year. Given the toxicity concerns and the impact on overall survival in randomized trials, it is important to look at the dose exploration in the selected dose of idelalisib. The approved dose for idelalisib is 150 milligrams BID or twice daily. As monotherapy, the maximum tolerated dose for idelalisib was not reached. Exposure response for efficacy plateaued at 150 milligrams. There was an exposure-response relationship for safety with higher exposures associated with increased toxicity, and this was coupled with high rates of treatment modifications due to toxicity. In combination, idelalisib 150 milligrams BID was also selected. There was limited dose exploration in combination, and again, there was no exposure-response relationship for efficacy, but there was an exposure-response relationship for safety. Ultimately, lower doses of idelalisib as monotherapy or in combination may have warranted further exploration. The second PI3K inhibitor is copanlisib, an alpha and delta PI3K inhibitor. The issues we will highlight are overall survival concerns in the Kronos 3 trial, PI3K-associated toxicity, and considerations for the selected dose. In 2017, copanlisib was granted accelerated approval for patients with relapsed follicular lymphoma who have received at least 2 prior systemic therapies. The approval was based on the Kronos 1 trial, a single-arm trial that showed an overall response rate of 59% with associated durability. The pooled safety database of 244 patients with non-Hodgkin lymphoma demonstrated a notable toxicity profile. there was a high rate of grade 3 or greater adverse events at 85%, serious adverse events at 51%, and high rates of treatment modifications due to toxicity. Since copanlisib inhibits the PI3K alpha isoform, it is associated with hyperglycemia and hypertension. The incidence of grade 3 or greater hyperglycemia was 34%. Grade 3 or greater hyperglycemia represents a blood glucose greater than 250 to over 500 milligrams per deciliter, with hospitalization indicated. For hypertension, the incidence of grade 3 or greater hypertension was 29%. Grade 3 or greater hypertension indicates the need for medical intervention. To mitigate risk, the toxicities of infection, hyperglycemia, hypertension, pneumonitis, neutropenia, and rash were included as warnings and precautions because they represented adverse reactions or safety hazards that are serious, clinically significant, and have implications for prescribing decisions or for patient management. In addition, a number of post-marketing requirements were issued for copanlisib. For accelerated approval, a post-marketing requirement was issued to verify the clinical benefit of copanlisib in patients with non-Hodgkin lymphoma based on an ongoing randomized trial. A total of 5 additional post-marketing requirements for safety were issued as shown. A supplemental new drug application for copanlisib was submitted in May 2021 for copanlisib in combination with rituximab for the treatment of patients with relapsed indolent non-Hodgkin lymphoma. The application was based on the KRONOS-3 trial, a randomized, placebo-controlled trial evaluating rituximab with or without copanlisib and a primary endpoint of progression-free survival. The population was patients with indolent non-Hodgkin lymphoma that included follicular lymphoma, marginal zone lymphoma, small lymphocytic lymphoma, and Waldenstrom's macroglobulinemia. The majority of patients enrolled had follicular lymphoma and marginal zone lymphoma. Importantly, the target population was those patients that did not require intensive therapy and were defined as either progression-free or treatment-free for 12 months or more following the last anti-CD20-based therapy, or considered unfit for chemotherapy due to comorbidities, and progression-free or treatment-free for 6 months or more following the last anti-CD20-based therapy. In the intent to treat population, the KRONOS-3 trial showed a statistically significant benefit in progression-free survival in those treated with copanlisib plus rituximab with an approximately 8-month improvement in PFS with an adjusted hazard ratio of 0.52. Here are the interim overall survival data for the KRONOS-3 trial. The Kaplan-Meier curve on the left is for the intent to treat population in indolent non-Hodgkin lymphoma, and the curve on the right is in patients with follicular lymphoma, which represented 60% of the trial population. It is notable that the copanlisib arm shows worse overall survival compared to the control arm within approximately the first 2 years. This is followed by a crossing of the curves, yielding the estimated hazard ratios less than 1. However, this pattern indicates a concern for potential harm early in the treatment setting with copanlisib in patients with indolent non-Hodgkin lymphoma who are suitable for treatment with single-agent rituximab. This table shows the reasons for death in the KRONOS-3 trial. The deaths due to adverse events were higher in the copanlisib arm and encompassed infections, respiratory, and cardiac causes. This graph shows the safety results from the KRONOS-3 trial. For grade 3 or greater toxicity, serious adverse events, and discontinuation, dose reduction, or dose interruption due to an adverse event, the rates were notably higher in the copanlisib arm, as indicated by the blue bars in the graph. The safety results from the KRONOS-3 trial demonstrate that the PI3K-associated toxicities of grade 3 or greater hyperglycemia, hypertension, infection, neutropenia, diarrhea or colitis, and any grade pneumonitis are driving the differences in safety between the treatment arms. Shown and indicated in the table, the incidence of any grade pneumonitis or grade 3 or greater PI3K-associated toxicities except increased ALT or AST, are substantially higher compared to the control arm. Based on ongoing analyses of the KRONOS 3 trial, the supplemental new drug application for copanlisib in combination with rituximab in patients with indolent non-Hodgkin lymphoma was voluntarily withdrawn from the FDA in December 2021. Turning to the selected dose of copanlisib, there are some important considerations. The approved dose of copanlisib is 60 milligrams IV administered weekly for 3 weeks in a 28-day treatment cycle. Notably, the 60 milligram dose was identified as the maximum tolerated dose, and there was limited dose finding in patients with hematologic malignancies. The PK and PD data suggested comparable efficacy at a 45 milligram dose and 60 milligram dose. There were high rates of treatment modifications due to toxicity at the 60 milligram dose level. Further, the 60 milligram dose was selected to be used in combination, and there was no dose exploration for copanlisib in combination. The third PI3K inhibitor is duvelisib, a delta and gamma PI3K inhibitor. The issues we will highlight are concerning overall survival, PI3K-associated toxicity, and dosing considerations. Duvelisib was granted regular approval for patients with relapsed or refractory CLL or SLL after at least 2 prior therapies in September 2018. The approval was based on the DUO study, a randomized open-label trial that demonstrated a statistically significant benefit in progression-free survival in those treated with duvelisib. The PFS results in the ITT population are shown in the table. The DUO trial enrolled patients who had received at least 1 prior therapy, but because of safety concerns, the indication was restricted to patients who have received at least 2 prior therapies. At the time of approval, the overall survival hazard ratio in those patients who had received at least 2 prior therapies was 0.82, favoring duvelisib. Duvelisib was also granted accelerated approval for patients with relapsed or refractory follicular lymphoma after at least 2 prior systemic therapies based on an overall response rate of 42% with associated durability from the single-arm Dynamo trial. The pooled safety database of 442 patients with non-Hodgkin lymphoma demonstrated a notable toxicity profile with duvelisib. There were high rates of grade 3 or greater toxicity at 84%, serious adverse events at 65%, and high rates of treatment modifications due to adverse events. The table on the right shows the grade 3 or greater PI3K associated toxicities and any grade pneumonitis for duvelisib, several of which included fatal and serious events. This graph shows the safety results from the randomized DUO trial in patients with CLL and SLL. For death due to adverse events, grade 3 or greater toxicity, serious adverse events, and discontinuation, dose reduction, or dose interruption due to an adverse event, the rates were notably higher in the duvelisib arm, as indicated by the blue bars in the graph. The safety results from the DUO trial demonstrate that the PI3K-associated toxicities of grade 3 or greater infection, neutropenia, diarrhea, or colitis, increased ALT or AST, rash, and any grade pneumonitis are driving the differences in safety between the treatment arms. Shown and indicated in the table, the incidence of any grade pneumonitis or grade 3 or greater PI3K-associated toxicities, except neutropenia, are 2 to 3 times or more higher in the duvelisib arm compared to the control arm. To mitigate risk, several measures were included as part of the initial approval for duvelisib. Similar to idelalisib, duvelisib included a boxed warning for infection, diarrhea or colitis, rash, and pneumonitis. Additionally, the toxicities neutropenia and hepatotoxicity were included as warnings and precautions. A risk evaluation and mitigation strategy was included with the initial approval of duvelisib to ensure its safe and effective use and that its benefits outweigh its risk. Along with risk mitigation, several post-marketing requirements were issued for duvelisib. For accelerated approval, a post-marketing requirement was issued to verify the clinical benefit of duvelisib in patients with relapsed or refractory follicular lymphoma. Additional post-marketing requirements were issued for safety that included characterization of long-term safety across ongoing trials and the final overall survival analysis of the DUO trial. As mentioned, FDA required the final OS analysis of the DUO trial be submitted as a post-marketing requirement. Here is the recent final OS analysis with a median OS follow-up of 63 months. The data for the final OS analysis is currently undergoing FDA review. The sponsor was required to conduct a confirmatory trial to verify the clinical benefit of duvelisib in relapsed or refractory follicular lymphoma. The DUETTO trial, a randomized trial evaluating duvelisib in combination with rituximab compared to investigators' choice of rituximab or R-CVP in patients with follicular lymphoma, was intended to be the confirmatory trial for duvelisib. The trial was never initiated due to feasibility issues and a changing treatment landscape. Because of the inability to provide evidence to verify the clinical benefit of duvelisib in patients with follicular lymphoma, as required per the accelerated approval regulations, the FL indication was voluntarily withdrawn in December 2021. For duvelisib, the approved dose is 25 milligrams BID. In general, there was limited dose exploration, and 75 milligrams was identified as the MTD. There was no exposure-response relationship for efficacy at 25 milligrams BID, and PD data showed near maximal suppression of the p-AKT biomarker at 25 milligrams. There were exposure-response relationships for toxicity between 8 milligrams and 75 milligrams, and the 25 milligram dose was associated with high rates of treatment modifications due to adverse events. Lastly, the fourth PI3K inhibitor is umbralisib, a delta PI3K inhibitor. Umbralisib was granted accelerated approval for patients with relapsed or refractory follicular lymphoma who have received at least 3 prior lines of systemic therapy, and for marginal zone lymphoma who have received at least 1 prior anti-CD20 base regimen. The approvals were based on the UTX-TGR-205 trial, a single-arm, multi-cohort trial that showed an overall response rate of 43% in follicular lymphoma and 49% in marginal zone lymphoma with associated durability. A subsequent supplemental new drug application for umbralisib was submitted in May 2021 for patients with CLL and SLL based on the UNITY-CLL trial, a randomized trial evaluating umbralisib in combination with ublituximab, the U2 regimen, versus obinutuzumab and chlorambucil in patients with CLL. Based on ongoing analyses and concerns with the UNITY-CLL trial, an FDA safety alert was issued in February 2022 for a possible increased risk of death in those treated with umbralisib. On April 15th, last week, the supplemental NDA for umbralisib and the BLA for ublituximab for the U2 combination regimen for the treatment of patients with CLL or SLL was voluntarily withdrawn from the FDA. This was due to updated overall survival data from the UNITY-CLL trial, which showed an increasing overall survival imbalance in favor of the control arm. At the same time, the sponsor announced the voluntary withdrawal of umbralisib from the U.S. market for the indications of relapsed or refractory follicular lymphoma and marginal zone lymphoma under accelerated approval. With that, I'd like to turn to our discussion of the PI3K inhibitor class and the topics as shown: a potential detriment in overall survival across multiple randomized trials, the differentiated safety profile of this class and how it impacts tolerability, dosing concerns with the selected doses, and the paradigm of single-arm trials. As a class, multiple randomized trials, as shown in the table, evaluating a PI3K inhibitor as monotherapy or in combination in patients with CLL or indolent non-Hodgkin lymphoma have shown a decrement or concerning overall survival compared to the control arm. Notably, the overall survival information from these trials is early and represents a low number of events. However, we are observing the same pattern repeated across multiple randomized trials. In addition, the trials show a favorable impact on efficacy endpoints, such as progression-free survival or overall response rate, indicating that the overall survival concerns are a primary safety concern. This is reiterated by the fact that a higher rate of death due to adverse events was observed in the PI3K inhibitor arms across these trials. It is also important to consider two additional components, the population of patients and the comparator arms. The patients are those with CLL and indolent non-Hodgkin lymphoma, diseases that have a long natural history. Progression isn't necessarily an indication for treatment, and patients have multiple effective treatment options with known efficacy and safety. For the comparator arms, they represent single-agent CD20 monoclonal antibodies or chemoimmunotherapy regimens, each with a favorable and tolerable safety profile, setting up an optimal comparative background to assess toxicity of the investigative PI3K inhibitor arm and its effect on overall survival. PI3K inhibitors have substantial toxicities that can be fatal or serious. The toxicities observed are driven by PI3K-associated toxicities related to the mechanism of action of these agents. This table shows the incidence of PI3K-associated toxicities for the approved PI3K inhibitors in patients with hematologic malignancies when administered as monotherapy. The incidence of the respective grade 3 or greater toxicities are notable and reiterate the overall safety concerns with this drug class. When looking at the overall safety results from the randomized trials evaluating PI3K inhibitors, each trial has shown higher rates of death due to adverse events, grade 3 or greater toxicity, serious adverse events, and treatment modifications. The differences in safety are driven by the PI3K-associated toxicities. Given the toxicity concerns with the PI3K inhibitor class, optimized dosing is warranted. The PI3K inhibitors exhibit a narrow range between an effective and toxic dose. Across the class, there has been limited dose exploration. For each approved PI3K inhibitor, there are exposure-response relationships for safety, primarily for PI3K-associated toxicities. Conversely, exposure-response relationships for efficacy have generally not been observed. Despite the need to balance efficacy along with safety and tolerability, there has been insufficient dose exploration as monotherapy and in combination for these agents. As noted, PI3K inhibitors have exposure-response relationships for safety, with higher exposure leading to increased risk for toxicity. These graphs show that with higher PI3K inhibitor exposure, there is an increased risk of diarrhea with idelalisib and umbralisib, as shown in the top left, an increased risk of infection, and specifically pneumonia with duvelisib, and an increased risk of hepatotoxicity with duvelisib and umbralisib, as shown on the right. This is in the setting that generally no exposure-response relationships have been observed for efficacy. Dose modification data from the approved PI3K inhibitors suggest tolerability concerns. Because of toxicity, a number of patients discontinue treatment or require dose reductions or interruptions. These graphs show the number of patients that receive each dose per cycle for idelalisib, duvelisib, and copanlisib as monotherapy at the currently recommended doses. As shown, a number of patients require treatment modification early in the treatment course, and many end up discontinuing therapy. This reiterates the need for adequate dose exploration and identification of an optimal dose. In addition, rigorous measurement of patient-reported side effects during dose finding or registrational trials allow for a better understanding of tolerability and toxicity. Information on patient-reported symptomatic adverse events were limited or not completed for the approved PI3K inhibitors. The last issue we would like to highlight today is the paradigm of using single-arm trials to support an assessment of benefit-risk for PI3K inhibitors, given the toxicity concerns noted. The prior issues discussed for the PI3K inhibitors highlight the limitations of single-arm trials. Foremost, without a comparator arm, it is challenging to characterize safety. The side effects observed could be due to the drug or to the underlying disease. Additionally, within a single-arm trial, the follow-up is often relatively short and characterizing long-term safety is limited. Second, the assessment of efficacy is less robust because comparison to a historical control or across populations has known limitations. Further, response rate may not predict clinical benefit. And finally, time to event endpoints such as progression-free survival and overall survival cannot be accurately interpreted in single-arm trials. In a single-arm trial, it is hard to balance the observed efficacy with toxicity to appreciate the true benefit risk of the drug in the intended population. As noted, the PI3K inhibitor approvals for patients with indolent non-Hodgkin lymphoma were based on single-arm trials and were accelerated approvals with a requirement to conduct a confirmatory trial to verify clinical benefit. The FL and SLL indications for idelalisib were voluntarily withdrawn in February of this year due to enrollment challenges in the ongoing confirmatory study. The FL indication for duvelisib was withdrawn in December 2021 as a confirmatory study was never initiated due to feasibility concerns and a changing treatment landscape. Last week, the umbralisib FL and MCL indications were voluntarily withdrawn based on concerns from a randomized trial in a relevant population. The withdrawals of the indications and the reasons for the withdrawals further highlight the limitations of the paradigm of using single-arm trials for development and potential registration of PI3K inhibitors. On the last slide, we discussed the limitations of single-arm trials. We would like to take a moment and highlight the benefits of randomized trials. Randomized trials are the preferred approach to evaluate a treatment and determine whether it provides clinical benefit. The act of randomization balances patient characteristics, both known and unknown factors, between the treatment groups, allowing attribution of any differences in the study outcome to the treatment being evaluated. This is not possible with any other non-randomized design. The act of randomization can also help reduce bias, including selection bias. Lastly, time to event endpoints such as progression-free survival and overall survival can be adequately assessed and interpreted in a randomized trial. The findings in the randomized trials of the PI3K inhibitors highlight the importance of overall survival information. While overall survival is not always feasible as a primary endpoint, such as in trials in CLL and indolent non-Hodgkin lymphoma, where progression-free survival is used as a primary endpoint due to the long natural history of the disease and multiple therapeutic options, overall survival is an endpoint that should be analyzed in all randomized trials. The FDA requires overall survival information for any trial that uses progression-free survival as a primary endpoint. Overall survival is an objective measure of clinical benefit and is considered both an efficacy and a safety endpoint. An evaluation of toxicity is embedded in an assessment of overall survival, including the ability to assess short-term and long-term toxicity. The same degree of statistical considerations that apply when overall survival is used as a primary efficacy endpoint do not apply when overall survival is evaluated as a safety endpoint. As mentioned, time to event endpoints such as overall survival can only be accurately assessed and interpreted in a randomized trial. Finally, overall survival is an important metric in supporting a benefit risk determination, especially in the setting of substantial toxicity. To end my presentation today, I'd like to review the evidentiary criteria that must be provided by sponsors to support approval. For safety, there must be sufficient information to determine that the drug is safe for use under the conditions prescribed, recommended, or suggested in labeling. For effectiveness, there must be substantial evidence of effectiveness that allows for the conclusion that the drug will have the effect it purports or is represented to have under the conditions of use prescribed in labeling. Ultimately, it is incumbent upon sponsors to provide evidence to the FDA to support that the drug is safe and effective in the intended population from an adequate and well-controlled trial or trials. So in conclusion, the PI3K inhibitor class has substantial toxicity, primarily related to the mechanism of action of these agents. The toxicity concerns have translated into a potential detriment in overall survival in multiple randomized trials in patients with CLL or indolent non-Hodgkin lymphoma, which is unprecedented in oncology. The PI3K inhibitors have tolerability concerns with high rates of treatment modifications due to toxicity. As a class, dose exploration and optimization has been insufficient, especially given the narrow range between effective and toxic doses. Finally, there has been a reliance on single-arm trials in patients with indolent non-Hodgkin lymphoma, limiting the assessment of efficacy and safety and precluding evaluation of the impact on time-to-event endpoints, such as overall survival. Therefore, the experience with the PI3K inhibitor class in patients with hematologic malignancies requires a re-examination of PI3K inhibitor development and the approach needed for sponsors to provide adequate evidence to determine safety and efficacy. We would like the committee to discuss the following. Please discuss the observed toxicity of the PI3K inhibitor class and whether randomized data are warranted with an assessment of overall survival to support the evaluation of benefit risk in patients with hematologic malignancies. The voting question, given the observed toxicities with this class, previous randomized trials with a potential detriment in overall survival, and a narrow range between effective and toxic doses, should future approvals of PI3K inhibitors be supported by randomized data? Thank you. This concludes my presentation.
This is DFO Shijia Chen. At this time, I would like to invite Dr. Anthony Song, an ODAC member, to please introduce yourself and state your affiliation into the record. Dr. Song? Thank you.
Hi, this is Anthony Sung. I'm an associate professor of medicine in the Division of Hematologic Malignancies and Cellular Therapy at Duke University. Sorry I had stepped away for a moment during the original introduction period.
Thank you, Dr. Song. Now I'll hand over to Dr. Garcia.
Thank you, Dr. Chen. We will now take clarifying questions for the presenters, the FDA. Please use the raise hand icon to indicate that you have a question, and remember to clear the icon after you have asked your question. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it would be helpful to acknowledge the end of your question with a thank you and end of your follow-up question with that is all for my questions, so we can move on to the next panel member. Dr. Nieva?
Thank you. This is George Nieva from USC. My question is for Dr. Richardson, and it relates to the safety of these drugs over time. It seems that the trial design for many of these studies was a treat until progression design. However, that is not the way that many indolent lymphomas are treated, where the patients are treated to best response rather than until progression. I guess my question is, are there data on duration of therapy and toxicity? And I guess the follow-up question to that is, are there any guidances from the FDA to sponsors as to whether or not there was a need for a treat to progression design? Thank you.
This is Nicole Gormley. If this question was directed to Dr. Richardson, I'll have him start, and then I will add on.
Hi, this is Nicholas Richardson, FDA. Thank you for the question. So I'll try to address them one at a time. So one, you had mentioned timing. So if you look at the safety of these agents when they are administered as monotherapy, the exposure in the pooled safety database that was submitted as part of the initial evaluation of safety that supported the approvals of these agents, we saw that the median exposure was typically 6 months or less. So specifically for idelalisib, it was a median of 6 months. Copanlisib was a median of 4.3 months. Umbralisib was a median of 5.9 months. And duvelisib was actually a little bit longer with a median of 9 months. And so for timing-wise, you know, we had a limited exposure when we initially assessed the safety of these agents. And so all the safety data that you are primarily seeing is really within sort of that first 6 to 9 month window. I will say that as part of the reviews for that, we also looked at the time to onset of a lot of the PI3K associated toxicities. And so based on the data that is in the respective labels for these agents, typically, actually I can just go through each one. So for instance, for grade 3 or greater diarrhea or colitis, the median onset was anywhere from 3 to 6 months across the 4 agents. For hepatotoxicity, that seems to be a signal that occurs typically earlier. So it's typically within the first 2 to 3 months of therapy if patients are going to experience hepatotoxicity. Pneumonitis was much more variable, where we saw a median onset typically around 4 months, but patients that had late onset all the way up to 19 months for pneumonitis. For patients that experience PI3K associated rash, typically it was within 2 to 4 months based on the data that is in the labels. And those are the main data that we have in regard to timing. So maybe I'll pause there before I get into the design consideration that you had mentioned regarding continuous administration versus something different.
Thank you.
Thank you. Dr. Chang, please proceed with your question.
Sorry, this is Nicholas Richards. Can I address his design comments? Is that okay, Dr. Garcia?
Absolutely, please proceed.
Yeah, so Nicholas Richardson again, FDA. So as far as design, you make a good point. So, you know, PI3K inhibitors are intended to be given continuously until progression or unacceptable toxicity. And a lot of the randomized trials that are noted, you know, utilized comparator arms that were typically fixed duration therapy. And so it's not a design that we encourage because we do acknowledge that there are differences when you are evaluating a continuously administered treatment versus a fixed duration treatment. However, the designs as they were conducted do allow us to have an appropriate comparative assessment of these two types of administration and do adequately quantify the risk. However, you know, it is not a typical design that we encourage, just given that the differences in administration can impact the interpretability of the results of the trial.
And this is Nicole Gormley. I'd like to just add on to that, and I think this is a really important point. When we talk about dose optimization, we are, including, optimization of the dose and, exposures, but also, looking at schedule and administration. And it's, quite conceivable that, the continued administration until progression, contributes to some of the, undoubtedly contributes to the toxicity that we're observing. And so when we're talking about dose optimization, it should be, the schedule should also be considered as part of that optimization to ensure that it's ultimately, tolerable and adequately safe for patients. Thank you.
Thank you.
Just to make sure, Dr. Nieva, did these comments address your questions?
Yes, fundamentally that's the issue here is the excessive toxicity for these agents built into this design of treatment to progression, but it sounds like the duration was not much longer beyond when maximum response would be achieved although for some of the agents was significantly longer than others.
Good. Well, we'll move on. Dr. Chang.
Thank you, Dr. Garcia. John Chang, industry rep. This is a question for either Dr. Gormley or Dr. Richardson. My first question is that, thank you for very nicely laying out the situation. But in the appendix, the idelalisib study in CLL, I think had an overall survival hazard ratio of 0.34 on table 15, I think. And so I'm interested in understanding how the FDA is viewing that result, which a relatively positive overall survival hazard ratio, although I appreciate the numbers might be small, in the greater context of a potential toxicity class effect risk, because obviously, you know, there was a withdrawal in other indications. My second question is a little bit trying to understand, I think you make the case for Project Optimus and the importance of understanding exposure response. My question is, does the FDA have a perspective on this dose optimization? Is it per indication or is it per agent? Because there are a number of indications within hematology, let alone outside of hematology. And so how one approaches a project, an optimization of the dose, is it specific to an indication and therefore has to be defined per indication, or is it per the molecule? I just would be helpful to understand the FDA's perspective.
Yes. So thank you for the question, and there are several questions there. You know, one about Project Optimus and dose optimization versus for an indication versus the molecule, and then thirdly, I believe you asked about the idelalisib 0116 trial. So I'd like to actually just, if it's okay first, I'd like to ask Dr. Brian Booth just to present a little bit about, you know, what we're thinking about and what we mean by dose optimization. Just to ensure that, you know, there's level setting here and we're all on the same plane as to what we're really talking about when we talk about the concepts of dose optimization. So Dr. Booth, would you mind just presenting a little bit about what we mean by dose optimization and also Project Optimus?
Certainly. Good afternoon. Can we please bring up slide 182? So again, my name is Brian Booth. I'm the director of the Division of Cancer Pharmacology 1 in the Office of Clinical Pharmacology at the FDA. Here we go. Thank you. So with respect to dose selection for oncology, we've generally pursued an MTD approach. However, we have many examples of oncology drugs with significant toxicities, including the PI3K inhibitors, that have required dose modifications or dose interruptions in the post-approval setting. So we need to reconsider our approach to dose selection and to think more about dose optimization for oncology drugs, especially with the current therapeutic options that are available, such as targeted therapies. To illustrate this thought, the figure on the left depicts the exposure response curve for cytotoxic chemotherapy. Given the mechanism of action, you can see that the curve for toxicity closely parallels the curve for efficacy. It's not possible to distinguish between the two curves. In this case, it makes sense to pursue the dose based on the MTD concept. This maximizes efficacy, although at the expense of managing toxicity. However, with targeted therapies, such as the PI3K or TKIs, the curves on the right are typical of the exposure-response relationships that we see. Generally, we see an earlier plateau for efficacy, followed by a more gradual later increase in toxicity. So pursuing the MTD approach with these types of drugs doesn't make sense, especially for drugs with longer survival, who require longer periods of continuous drug treatment. In these settings, managing the toxicity of the drug has much greater significance. Next slide, please. But what are the implications of these exposure-response relationships for dose optimization? Generally, we maximize efficacy before toxicity. This especially is true if we use an MTD approach. The efficacy is plateaued, and increasing the dose further does not result in any further improvement in efficacy. In this context, we can say that the exposure response for efficacy is flat. However, the exposure response for toxicity is still in the rising portion of the curve, and changes in dose can impact the rate and severity of adverse events. In these situations, we may be able to reduce the dose without impacting efficacy significantly, while reducing adverse events. Next slide, please. So recently, the Oncology Center of Excellence launched Project Optimus. This project was initiated based on the recognition that many oncology drugs require dose adjustment and may lead to suboptimal therapy. The mission is to find doses of oncology drugs that maximize efficacy and tolerability. And one of the specific goals is to leverage the non-clinical and clinical data to better select these doses. Next slide, please. So in this slide, the traditional approach to dose selection in oncology based on the finding of the MTD is depicted. Generally, there is a dose escalation trial designed to identify the dose of the DLTs and subsequently the MTD. The MTD is then used in subsequent registration trials, which may be randomized controlled trials or frequently single-arm trials that are part of the accelerated approval pathway. With the MTD approach, the assumption is that higher doses will have higher efficacy, and which then maximizes the efficacy at the expense of toxicity. Generally, a 3+3 design is used, so there are a limited number of patients to assess the pharmacokinetics, the pharmacodynamics, safety, and efficacy at each dose level. And the observation period to assess DLTs and toxicities is often too short to obtain useful information on dose modifications, including dose interruptions, reductions, attenuations. Next slide, please. In contrast, a dose optimization strategy like the one depicted here has a higher chance of identifying the dose with a benefit risk. We begin with the same dose escalation design, but with the purpose of better understanding the pharmacokinetics, pharmacodynamics, safety, and efficacy at each dose level. This will often include dose expansion of several cohorts to generate these additional data at promising dose levels. Further, in this paradigm, longer periods of observation are incorporated to assess adverse events, including the onset of delayed toxicities, in contrast to the MTD approach. Next slide, please. Additionally, for dose optimization, there are some more specific recommendations that should be evaluated in order to better select the dose or doses for development. Consideration should be given to non-clinical data such as in vitro or in vivo receptor occupancy or enzyme inhibition, because this provides support that the concentrations of the doses selected are in the right range. In early trials, sufficient PK sampling in a sufficient number of patients is necessary to adequately characterize the pharmacokinetics in order to understand the behavior of the drug and any PK limitations such as saturable absorption, and to develop exposure-response relationships. This data is also important in identifying exposure-response relationships with biomarker data, as well as with the safety and efficacy data in early trials to better assess optimal doses. These exposure-response relationships can also be used to predict patient outcomes by dose level, which can also aid in selecting doses for development. Unlike the MTD approach, there should be some expansion of several promising dose cohorts with sufficient numbers of patients to better understand and evaluate the PK, safety, and the efficacy. Another important approach that should be considered is to conduct randomized parallel dose-response trials, which ensures similarity of patients at each dosage and aids in the interpretation of dose and exposure-response relationships. Finally, another possibility that can be considered is to include multiple doses as part of the registrational trial. Next slide, please. With respect to dose optimization combinations, this can get quite complicated, but the following are some general recommendations to consider. First, simply taking the approved monotherapy dosage and applying it in combination with another drug is likely to cause excessive toxicities, and some dose exploration is warranted. When combining two new drugs, a thorough understanding of the PK, PD, safety, efficacy, and the exposure-response relationships for safety and efficacy for each drug should have been assessed as prescribed for monotherapy. For two new drugs, there should be dose exploration with different levels of each drug. It may be appropriate to conduct more dose exploration in these combinations with a drug that appears to be more active or more toxic. If a new drug is to be added to an add-on therapy, it may be appropriate to consider some dose exploration with the established regimen in addition to the new drug. Another point that should be considered is that in the combination setting, smaller dose increases in those tested in monotherapy dose escalation trials should be assessed. As with monotherapy, exposure response curves for safety and efficacy should be evaluated in each drug in the combination. And lastly, occasionally drug-drug interactions occur between two drugs in the combination, which can result in higher exposures than anticipated, particularly at steady state, which may cause unexpected or unwanted toxicity. The dose exploration optimization studies should also provide a provision for assessing the DDI liability of the regimen. Thank you.
Thank you. So this is Nicole Gormley, and I think we've sort of already touched a little bit on your second question as well about whether or not dose optimization is indication specific or if it's molecule specific. And really, it's a little bit of both. For dose optimization generally, we should always be incorporating information gleaned from earlier stages. And what I mean by that is, oftentimes we'll start in a broader, population, initial dose escalation and dose finding. But then, once specific indications are identified, there may need to be dose escalation and optimization that's conducted separately. For example, a different dose may be optimal for an AML population that may be very distinct from what's needed in an indolent follicular lymphoma population. But hopefully, again, when new indications are explored, information from prior studies should be incorporated into those, dose optimization studies. And then, again, though, if you're looking at follicular lymphoma versus marginal zone, there may not be that many differences needed. And then the other aspect that comes into this, which Dr. Booth already touched on, is just when these products are then studied in combination, the need to again really make sure that the various aspects are considered, and that separate really dose optimization is needed when looking at something as a monotherapy or in combination for a new therapy or a new indication. And I'd like to make sure that we address the third question, which was about the 0116 study. So I'll ask Dr. Richardson to comment on that.
Hi, thank you for this is Nicholas Richardson, FDA. Thank you for the question regarding the idelalisib 0116 trial. Just as a refresher, this was a randomized placebo-controlled trial looking at idelalisib plus rituximab versus placebo and rituximab in patients with relapsed CLL, and this is what supported the approval of idelalisib in combination with rituximab for these patients. So for the overall survival information, you are correct that the overall survival hazard ratio for this trial showed a favorable effect, favoring the idelalisib arm with a hazard ratio of 0.34. As far as the interpretation, the OS information, as you noted, was still a limited number of events, and it was very early information. And the trial was terminated early following the statistically significant impact on PFS. So we didn't have longer-term follow-up to ultimately assess the overall survival information. But when we did evaluate the data that was submitted, it was actually very unclear as far as what factors accounted for the difference in overall survival. And just one note is patients on the placebo arm were able to cross over and receive treatment with idelalisib following progression, which does impact the assessment of overall survival. So it's a little bit unclear as what accounted for the difference in overall survival in this trial. And interestingly, it's a little bit of a standout compared to some of the other trials that we had discussed during the presentation.
So thank you for that. Yes, that does answer my question. I just did want to kind of make an aside. I appreciate the helpfulness in optimizing each dose or each indication combination. However, that does take time and resources, and so those are just factors to at least be aware of.
Thank you, Dr. Chang. We'll move on. Dr. Tanarajasingham, please, your question. Dr. Tanarajasingam, you may be mute. Please unmute.
Sorry about that. I have two questions. The first is about toxicity and tolerability, and the second is unrelated and is about these drugs addressing an unmet need. So Dr. Richardson, in answering Dr. Neva's questions, you had outlined some information about the timing to toxicities that's very difficult to find published or systematically reported anywhere by sponsors of the FDA. We know from clinical experiences that these PIK3 kinase inhibitors have short and long-term toxicities, and some of the immune-related AEs can be delayed, and when these agents are chronically administered, even low-grade AEs that are protracted can affect tolerability. And so when you're talking about dose optimization, do you think that going forth in drug development, sort of related to the presentation that was just given that was really helpful, with this class of agents, do we need longer dose-finding studies or DLT windows that include patient-reported outcomes, which we know are needed to understand tolerability? And I guess my question is, does the FDA have the authority to require these types of studies and also require the reporting of not only high-grade AEs, but the timing of these key lower-grade AEs that may be impacting tolerability, and require high-quality PRO studies assessing multiple domains of tolerability in this setting? It just seems like this is very important complementary information to the traditional survival outcomes with this particular class of drugs. That is the question about toxicity of tolerability, a lot packed in there, but the second one is a bit more straightforward. Of the trials you discussed, we're looking at trials in multiple biologically distinct disease groups and at times in different lines of therapy, different populations. To address a question of whether these drugs address a high unmet need in CLL, to your knowledge, and I suppose to my other colleagues on this panel, has there been any trial of PIK3 kinase inhibitors in patients with double refractory CLL, that is, those who have progressed on a BTK inhibitor and venetoclax? Thank you very much.
Hi, this is Nicole Gormley. I'll ask Dr. Richardson to respond. Thanks.
Hi, Nicholas Richardson, FDA. So thank you for the question. So I'll try to, I'll address the tolerability question first. So one thing you had mentioned is just, you know, I think in early phase trials, how do we sort of look at toxicity and tolerability? And, you know, given the lessons learned from the PI3K inhibitors, you know, how can we improve that process? And you had mentioned several things. So one is, you know, in early phase trials, a lot of times we identify a dose to carry forward based on an evaluation of dose-limiting toxicity. And you had specifically mentioned the DLT window. And typically a DLT window is one cycle, you know, and that is reasonable when you typically have a drug that is administered, you know, daily or twice a day, or, you know, for instance, for copanlisib, it's once a week for 3 weeks with 1 week off, to really sort of get a sense of dose-limiting toxicity. However, that does not incorporate the assessment of later onset toxicity, which we do see with these agents, and specifically some of the immune-mediated toxicities that are grade 3 or greater, we know can have a later onset based on some of the data that we have, even up to 6 to 9 months. And so one thing that is something that we do try to work in collaboration with the sponsors is the way that the totality of data is evaluated to really look at these early phase trials and the data generated regarding safety, PK, PD, exposure response, and preliminary activity. And so if we do have data, or if there's previous data with a same-in-class agent that indicates that later onset toxicity is a concern, typically we try to at least have a proposed plan of how that will be captured in the assessment of safety in these early phase trials. So it is a consideration and something that we do try to encourage sponsors to incorporate when they're really looking at all the available safety and PK and PD information when they are selecting a dose to carry forward. And then you had mentioned patient-reported outcomes and whether that should be included. And then just give me a second while we pull up a slide. So as part of an initiative within the Oncology Center of Excellence, we spend a lot of time and effort really looking into how the patient voice and the patient experience can be incorporated in all aspects of drug development. And so in early phase trials, we do recommend and encourage that patient-reported adverse events be incorporated because it does help inform tolerability. And you do have the ability to assess these outcomes even in early phase trials and even in registrational trials to really get a sense of tolerability from the patient standpoint. And there's been a lot of work done on the different measures of assessment that can be incorporated in these trials for the particular population that is being evaluated. And it's something that we encourage in all aspects of drug development. So I'll pause there and just see if any of my other FDA colleagues have any comments before addressing the double refractory CLL population and PI3K inhibitors.
Yes, thanks. This is Nicole Gormley. I would just add on, your question was specifically, as to whether or not we had the authority to require, further or more aggressive dose optimization or authority to require patient, generated data or patient-reported outcomes. And the answer, short answer is no, we do not. I will say, though, as was pointed out, excuse me, that was pointed out earlier by Dr. Cheng, dose optimization does require additional resources and it does require additional time. In our experience, though, and what we're seeing here in the PI3 kinase inhibitors and in other areas, is that it's time well spent. The investments that are made in finding the right dose, they improve outcomes for patients, and then it results in a better product in the end that allows us to, have confidence in the results from these studies. So while it's not something that's, within our authorities to require, it's something that we strongly recommend and encourage. And then also, just as well related to the patient-reported outcome information, it's crucial to have this information about how these products, impact patients. And, it's helpful to collect this information early, but it's also most robust when it's captured in randomized trials. And so we encourage sponsors as well to capture this information again. But again, we don't have the authority to require it. Thank you. I'll turn it back to Dr. Richardson to address the CLL question.
Hi, Nicholas Richardson, FDA. So thank you for the question regarding double refractory patients with CLL who are refractory to a BTK inhibitor or a BCL2 inhibitor. So as you know, BTK and BCL2 have changed the treatment landscape for CLL. And so, the development, at least of the approved PI3K inhibitors, was really sort of prior to the treatment landscape or in conjunction with the changing treatment landscape. So, we do have limited data at this time. However, there are ongoing development of products in this class, and there is clinical trials that are currently underway that do allow these patients to enroll, just given that they have failed two therapies that have known efficacy and safety and some trials showing a survival advantage.
Thank you very much to both of you. I appreciate the responses.
Thank you. We move on. Dr. Advani, please state your question.
Thank you. This is Dr. Advani from Stanford. So, to anyone from the FDA, most of these were global trials. So were there geographic differences in overall survival? Like, was it all across the board, or was it in underdeveloped areas? And the second question is, how many of these were recent in the last 2 or 3 years, where all these trials are reporting out and related maybe to the pandemic and the supportive care differences in different parts of the world?
Thank you. Thank you very much for the question. I'll allow, have Dr. Richardson answer this question. Thank you.
Hi, Dr. Advani. So thank you for the question. So I will freely admit I don't think we have a very at least data-driven answer for you. We'd have to go back and look. So, we typically do look, as far as a sensitivity analysis based on region, typically for the primary outcomes of, at least in the randomized trials of PFS and OS. Within a single arm trial, we do, in addition, look at region based on response rate. As far as specific data points, I'd have to look it up for you, but there have been no, overall regional sort of differences that would prompt concern regarding differences for the U.S. versus outside of the U.S. Although I will say some of the trials, just given the comparator arms that were chosen, some of the trials did have an imbalance where there was the majority of patients enrolled ex-U.S. And a limited representation of U.S. patients.
This is Nicole Gormley. Just on the first part of this question about the global aspects. Just another consideration here is that as we've highlighted, these analyses that we're talking about are really early, and several of them, and so it's hard to do additional sub-analyses based on the overall survival based on those results. When we look at toxicity, though, overall, and especially as Dr. Richardson, highlighted, we do analyses to look to see if there's regional differences or things like that. And again, there's nothing that grossly stands out related to that. Perhaps I'll start with the COVID question. And some of these trials were conducted before the COVID pandemic, and so we're not impacted. And that's the case with the vast majority of these trials. Some of the trials have been conducted more recently with COVID, where COVID could have had an impact. And I will highlight, though, that what we're talking about here are randomized trials. And so, even though COVID may be occurring during the time frame of the clinical trial itself, randomization should, control for any, imbalances or differences, things like that. So, we feel confident that, this is, not just a finding that's related to, the underlying COVID pandemic. And then, as mentioned, most of these trials were conducted before the COVID pandemic started. Thank you.
Thank you.
Okay, we'll move on. Dr. Kraft, you could raise your hand.
Walter Kraft. I had put my hand down because Dr. Booth had addressed most of the issues, but I will ask specifically about biomarkers. This is about a specific class of medications. For dose optimization, is there a biomarker that could serve as a surrogate towards a clinical endpoint that would optimize or help with the dose optimization across these drugs within the single class?
This is Nicole Gormley. I'll start the response and then open it up to other FDA colleagues if there's others that want to chime in. But, I would say that, in terms of biomarkers for, endpoints for, response assessment, that has not been uniformly developed across the class. There may be other markers that are helpful for dose optimization that could be looked at, et cetera. But again, that it's not, to my knowledge, there's nothing that's been uniformly done. I don't see.
Thank you.
Yeah, thank you.
Great. Next question is Dr. Out.
This is Jessie L from IQSP. I have a question regarding toxicity for this class of agents, especially in the context of dose optimization, and particularly for the combination therapy. And I think Dr. Booth probably and Dr. Richardson probably can help me here. So when I look at the data that Dr. Richardson presented today, as well as the briefing materials that were sent to us earlier, what is clear is for the single agent data, idelalisib, and when I compare that to the data in the randomized trial in the combination therapy, there seemed to be a very substantial pharmacokinetic interaction in the sense that the AUC that I saw on those graphs were very, very different, like 200% higher in the combination therapy, even though the combination therapy was using a lower dose. And secondly, at the same AUC level, the toxicity to the GI was, again, almost twice as high. So not only, and I'm wondering if I'm reading the data correctly, and if I do have read it correctly, then is this something that's common for this class of agents? Because if it is, then this class of agent may be teaching us a bigger lesson, and that is this agent, because of the mechanism of action and many downstream effects, may be actually causing very substantial PK/PD interaction on the host tissue level. Which means, for this class of agent will be a real big problem for when you develop a combination therapy and not do the dose optimization. So my question is, number one, am I reading the data correctly? Maybe Dr. Booth can help me there. And number two, would that be the same conclusion? And how do we deal with agents such as this? Because it has such a broad mechanism of action, many downstream effects, and when you're seeing PK/PD interaction at such a high level, how do we deal with it from the dose optimization standpoint?
This is Nicole Gormley. Thank you again for your question. I will ask Dr. Booth to respond.
Hello, good afternoon again. So I'm not quite sure what you're specifically referring to, Dr. Al, in terms of the data, but one of the concerns that I brought up earlier, and we sometimes see, is that when you put things into combination, you can end up having a drug-drug interaction. And that can raise the exposure of one of the drugs. And if you don't look at the situation long enough, you may not be aware of that. You can end up having exposures that are higher than anticipated that can confound or lead to these unwanted toxicities later in therapy.
Yes, I'm sorry. I was referring to the figure 4 and figure 9 in the briefing materials. One shows the single agents PK, and the other one shows the endotoxicity, and the other one shows the combination therapy. And what caught my eye was the much, much higher AUC in the combination therapy, and a much more severe toxicity even at the same AUC. But I think you're right, there's probably a DDI going on, but I don't know how the PD interaction becomes so severe. But I cannot refer you to the slide because figure 9 was not on the slide. Figure 4 was on slide 49 of Dr. Richardson's presentation. On the left panel, right? That's the single. This is the single agent plot. But if you go to the combination therapy plot, which is not on here, you'll see double the AUC, and then not only that, the probability for toxicity at the same AUC become twice as severe, which to me suggests PK/PD interaction, on both the effect level and on the kinetic level.
Right. I think we're on slide 133.
This is the combination, this is the combination. So if you look at the same AUC, about 15,000 units, which is the one on slide 49 for single agent, same agent, it was 20% probability, now it's about 40% probability. So somehow, it could be the delay effect you're talking about where the immune system is working, acting up as well. But you're seeing at least additive effect, I think, with the other combination agents.
Yeah, potentially. I'd also like to invite.
Yeah.
I'd also like to invite Dr. Leanne Ma from our pharmacometrics group to see if she has any additional thoughts on these analyses.
Hi, this is Lian Ma from Pharmacometrics FDA. Yeah, another potential explanation for the difference in the exposure scales, it could be that the exposure metrics is slightly different. For the monotherapy plot, the exposure I think is related to AUC for the dose 1, and for this one, it seems to be AUC within the 24 hours. So there might be a slightly difference in terms of how to derive the AUC metrics. But again, I think I agree with your comments that the substantial increase or difference in even the toxicity rate could be partially due to the overlapping toxicity between the two agents in the combination.
Thank you. I think what this database said to me is this class of agent is very special, and it will help me on my vote. So thank you very much.
Thank you.
Thank you both, the FDA and Dr. Out. Moving next to Ms. Nadine Baker.
Thank you. This is Michelle Nadine Baker, and I have a follow-on question to what Dr. Gita was asking, and that is, I know that in both Dr. Gormley's and Dr. Richardson's presentations, they talked about there being a variety of options for patients of drugs, specifically CLL and SLL patients, to take. But within those, once patients develop resistance, or perhaps if they have comorbidities, those drugs are not viable options. I do realize that some, but not all, mention that they need to, that a patient to go on one of these needs to have 2 or more previous treatments, but I don't see that across the board on all of them. Is that something that would be made specific to this class of drugs in the future? And therefore, they could still be used as an option when patients run out of others, sort of a clinical trial?
Thank you. This is Nicole Gormley again from the FDA. Thank you very much for the question. And I think, you bring up a really important point. And this is something that, we spend a lot of time thinking about, because, you are absolutely right. Patients do relapse, or unfortunately sometimes, develop refractory disease. And there is a need for, additional therapies for these patients. However, when we're in that sort of situation, it's really important that the drugs that patients receive, even if they have exhausted all of their therapies, that we know that these are, safe. And so, we would not be in a situation where we would just, change the indication for a class of products if we didn't have data in that population. And then just to answer, thank you for displaying the slide. Just to answer, your question a little bit about some of those have different lines, et cetera. And, some of this is just a temporal factor here, in that, these were approved over various time points, and then, different therapies became available. We're talking about, quite a time span from the approval of the first one to the last one. So some of these are just represent, changes in treatment landscape during that time. But I guess I would underscore, though, that, when we're looking for, therapies for patients, we still need to have confidence that they are safe and effective, and we would not, adjust an indication without having data to support that. I hope that answers your question.
Thank you. And I have an additional question, which is regarding things like on page 14 of Dr. Richardson's presentation, when there are things added on PMRs, such as to the regarding a drug, such as a Dear Healthcare Provider letter and boxed warnings. Does the FDA provide any oversight on how prevalent these communications are? And is there any education that's also a requirement of the company for the providers and patients to ensure that they are understanding that, I mean, they're reading these, that they actually get to them and they're understanding, for this is more for community physicians, what these mean?
Thank you for your question. And I guess I would just start by saying I think you bring up a very important point. And, you know, in terms of specifically how is information disseminated to providers. So we have, you know, we have, when a drug is approved, we issue and include information in the prescribing information or the label, the PI, about, you know, the safety risks, et cetera. And, you know, in certain instances, when there are significant safety findings, we will include a warnings and precaution or a box warning. Several of these products were approved with a REMS, which a communication REMS, which, and I can't, you know, provide the specifics of what was included within the REMS at this time, but, you know, they include information or require that there be communications to prescribers, and there is ongoing assessment of that communication as part of the REMS. So those are actions that can be taken at the initial, you know, time of approval. So when we were made aware of some of the findings, for example, with idelalisib, the FDA issued a safety alert, and that goes through various channels. It's placed on the FDA website. And there are distribution lists to providers and medical professional societies associated with that to make them aware of new safety findings that we become aware of. So often then sponsors will issue a Dear Healthcare Provider letter that goes along sometimes with the FDA safety alert, or can be issued on their own as well. And we think that it's really important that these Dear Healthcare Provider letters are issued broadly to providers, and again, medical societies, et cetera, to ensure that that information is widely communicated and available to providers. And we engage with sponsors to ensure that that happens, but it is within the sponsors, you know, purview, so to speak, to figure out initially, anyways, the distribution. And, you know, if we feel that the distribution is not adequate, we will or can pursue other avenues for FDA then led communication. But I think it's of the utmost importance that providers and patients be aware of safety findings with these products.
Thank you. I have no further questions.
Thank you. I know Dr. Diel, Dr. Cristofanilli, and Dr. Dunleavy have a few questions. Maybe in the interest of time, for us to stay on track, we can actually save those questions for our section after the OPH session. So please, Dr. Diel, Cristofanilli, and Dunleavy, just bear with me, hold those questions for a little bit later, and I promise you we're going to start with the three of you during that session. We will now take a quick 10-minute break. Panel members, please remember that there should be no chatting or discussion of the meeting topic with anyone during the break. We will reconvene at 2:15 p.m. Eastern Standard Time. Thank you. Okay, we're going to go ahead and start again. I would like to state into the record that no one registered to speak for the open public hearing session. We will now take remaining clarifying questions for all the presenters. Please use the raise hand icon to indicate that you have a question, and remember to put your hand down after you have asked your question. Please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. As a gentle reminder, it would be helpful to acknowledge the end of your question with a thank you and end of your follow-up question with that is all for my questions, so we can move on to the next panel member. So just to get back to the three pending questions that we have from the earlier session, we're going to go with Dr. Diel, if you don't mind just to ask your question.
So Lou Deal, Duke University. Dr. Booth talked about in dose optimization about a randomized trial, which immediately begs for me the second question, which is how do you select the lower dose? We know how to select the upper toxic dose, but how would you select the lower dose? Second part of the question is there enough information in the phase 1 trials that would tell us where toxicity starts and where efficacy starts to actually make a guess at what that second dose would be?
Great. Thank you.
End of the presenters, yes.
Okay, great, thanks. I'll ask Dr. Booth to start the response. Thank you.
Hi, thank you for the question. So I think, in my presentation, I listed a number of bits of evidence that we can rely on to help make some decisions about, what sort of doses to look at. So some of this would be some of the in vivo stuff, some of the interactions with like something like the PI3K inhibitors or receptor occupancy, that sort of thing, give us some indication of whether we're in the right ballpark. We could also use some of the PK/PD information that comes out from some of the early trials. So for instance, let me go to slide. Let's try slide 139, just as an example. So this is for duvolesib. So they had these ex vivo models where they were looking at the effect of the different doses and concentrations on the PAKT suppression in their cell models. Okay, and so you could see very early on that the 25 milligram gave them almost complete suppression of this biomarker. Okay, and so that helps us to better understand what sort of doses in vivo are going to be in the right ballpark. Okay? Then further, you'd have to look at what you see in the early clinical trials in terms of the safety and the efficacy that comes out of that, and evaluate all of that to make some decisions about what sort of doses you're going to take forward. And I would invite others to chime in on that if they have other things to add.
So I guess the other part of my question is, does that actually correlate with outcome, either toxicity or efficacy?
Well, to some extent it certainly does. So we know that duvolesib seem to have some activity at that level. It's not, you can't use one single piece of the information to make that decision. I think you have to look at this in terms of the collection of information and the different things that you've looked at, including the in vitro and the non-clinical, as well as the early data from the trials that you get.
And this is Nicole Gormley. I think I'd like to just add on. I think, we find ourselves, in a situation where we have some early data to suggest that for several of these products that lower doses may have been equally effective. But, the issue is that we find ourselves with across the class generally is that, we don't have lots of data or robust data at the lower doses. And I think that's where a randomized dose-finding trial or a randomized phase 2 trial could really be helpful. Spending a little bit more time at dose optimization where you're collecting more robust data at lower doses compared to the higher doses for both efficacy, safety, tolerability, patient-reported outcomes in a randomized dose findings setting, then you'd be well prepared to go into a trial for registration, having confidence in the dose that's been selected. And then we would know definitively whether or not that lower dose or whatever dose the best optimized dose is, in the randomized trial, what the efficacy and safety and ultimate clinical benefit would be from that randomized trial that would then be used for registration.
I don't want to play the devil's advocate, but would you really know that if you only selected 2 doses to use?
I mean, you may never perfectly know that you have the best dose possible, but I think we would know that we would have, more information than what we currently have. So, I think it would have to be informed, by the early initial phase 1 dose finding, et cetera, and I think you can have inklings then or more information if you had a randomized phase 2 and again, selecting the winner, so to speak, but then also gleaning more information. And I guess I would just, add that, dose optimization really can be done, throughout a drug development course, looking at the initial phase 1 trials, and then, randomized phase 2 trials where you're gathering more information. And then even in the, phase 3 trials, additional aspects or things can be done to make sure that you have the most optimized dose, making sure that the schedule and other supportive medications, et cetera, are adequate. So I think, that this can be paid attention to. And you're right, at some point we may never know that we have the best dose, but I think we'd be in a much better situation than what we are now.
Yeah, thank you very much. That relieves my mind a little bit on the question.
Thank you. We move on to Dr. Christopher Nally, please.
Yeah, hi. This is Dr. Christopher White Cornell. And I have a question for Dr. Richardson. You did a great presentation and went over the toxicity and efficacy of the studies, as well as remind us that the overall survival is an objective endpoint. As you know, many times it's not only a matter of toxicity affecting the overall survival, but also the subsequent therapies and the ability to really continue treatment with efficacious agents. So we have seen that this indolent disease should have a number of different options. So do you have any information in the randomized studies about the number and the type of therapy that these patients receive after they were off the drug or they progressed? Because it seems to me that it may certainly be an issue related to the dose, but it could also be an issue with this class of drugs with a specific target, may affect bone marrow, may affect other liver function, at the point that you are unable to continue treatment when it is a chronic administration and is also affecting the administration of the other therapies. Thank you.
Hi, this is Nicole Gormley again. I want to make sure I understand and make sure we adequately address your question. Your first question was sort of, and correct me if I'm wrong, that that it's not necessarily easy to ascertain what the cause of the overall survival findings may be, that it may be due to toxicity.
Yeah, I think in general, the overall survival, when you have one intervention, is due to a number of factors, and not only one therapy, but the accumulation of therapy that the patient receives, particularly in an indolent disease, right? So 5 or 6 line of therapies before or after the agent that be studied. But if you do have an agent that affects the ability to receive subsequent therapies or affect the efficacy of subsequent therapies, then you have another issue that you have to deal with. So do we know how many therapies, which type of therapy this patient received after they complete or the treatment either for toxicity or for progression?
Yeah, yeah. And I think, first I'll just mention just briefly about the overall survival comments. This is Nicole Gormley again. You are, absolutely right. And that, overall survival, it's really an assessment of multiple things. And overall survival, can be, impacted by inadequate dose, perhaps, and too many toxicities. It could be just related to the toxicities of this class. It also could be impacts on patients that result in inability to tolerate subsequent therapy or inability to respond to subsequent therapies. And so, overall survival really is a assessment of multiple factors and impacts of the drug. And I'll ask Dr. Richardson, to comment about, your other question in terms of, subsequent therapies that patients may have received after participating in the clinical trial. Dr. Richardson.
Yeah, hi, Nicholas Richardson, FDA. So as Dr. Gormley mentioned, this is a good question, and as we mentioned, for just using the PI3K inhibitors as an example, overall survival, we look at from an efficacy and a safety standpoint. As we went through in the presentation, nearly all these trials had an advantage in PFS or overall response rate, which, really sort of makes us focus on, the safety aspect of overall survival. And one thing that you mentioned that's really important in the setting that we're talking about today, patients with CLL or indolent non-Hodgkin lymphoma, is, they are considered indolent diseases, and patients do have the ability to receive multiple subsequent therapies. So when we do look at overall survival, to really sort of address the impact of subsequent therapy, we go about it looking at, what death events occurred, while on therapy or within typically 30 days within the last dose of therapy. So essentially a treatment emergent event. The other thing that we look at is whether there is a temporal association with the fatal outcome. And as just an example, we know that some of the PI3K inhibitors cause dermatologic toxicity, and so, we've had cases where patients have experienced either a grade 3 or grade 4 sort of rash event, and then that rash subsequently became infected, and ultimately the patient succumbed due to infection. And so if there's a temporal relationship, we also consider that when we are evaluating the reasons for death. To address subsequent therapy, we do ask sponsors to provide us information regarding the timing of subsequent therapy. And if available, we do request information on the specific types of subsequent therapy, so we can get a sense of just what you mentioned. Does the therapy that's being evaluated in the trial, either impact their ability to receive subsequent therapy, or does it impact the ability to respond to subsequent therapy? And as you mentioned, PI3K inhibitors are immune modulators, and so there is a concern, in regard to subsequent therapies and how patients may respond. So we do evaluate all of that to really try to get a sense and fully characterize the overall survival information from these trials.
Thank you.
Thank you. Dr. Dunleavy, do you have a question? Dr. Dunleavy, you may be mute still.
Hear me now, okay?
All right, let's just move to the next question. Dr. Conaway?
Yes, I'd like to express how important I think Project Optimus is and the need for dose optimization. I think really the central issue here is the choice of dose that went into some of the early studies. At Virginia, we have a Center for Early Phase Trials that's researching, advocating, implementing dose optimization designs, much along the lines of what Dr. Booth laid out. And I wanted to point out in response to some of the questions, there are statistical designs to handle nearly all of the issues that were discussed today. There are designs for evaluating both safety and efficacy for targeted agents with curves that were depicted by Dr. Booth. There are designs for combinations of agents to explore the surface, multiple combinations of agents. There are designs for late onset toxicities, heterogeneous groups of patients, designs that will incorporate patient-reported outcomes, PK data, biomarkers, or to allow you to investigate multiple schedules. So I think that the technology really does exist to do greatly improved dose optimization. So I think that that's an important thing that we should be aiming for. So that's a general comment. My specific comment is, in answer to an earlier question, there's apparently no authority for the FDA to mandate designs, but one concrete suggestion I would like to see is perhaps recommending or requiring operating characteristics of whatever design is used, some measure or some quantification in the degree of uncertainty in the dose that was actually selected, or those calculations or simulations might lead to a recommendation to move more than one dose forward. So I think even if whatever design is proposed, I think that some degree of quantification of the uncertainty in the results of that design would be very useful. Thank you.
Thank you, Dr. Conaway. Maybe we'll go back to see Dr. Dunleavy.
Yeah, I apologize about that. So I also had a question about toxicity. With this class of drugs, when you see immune-related toxicities and the immune-related toxicities that have been reported in clinical trials, you're particularly struck by the unpredictability of onset of those toxicities. And the question really is how are host and disease-specific factors interacting with dose-specific factors in causing those toxicities. I mean, there have been some observations with this class of drugs that immune-related toxicities are different in different patient populations. They may be different in different age groups. We've been talking about follicular lymphoma and CLL, so there are some differences if you look at different classes of agents in those diseases in terms of their biology. And I guess the question is in the data that we have so far with PI3 kinase inhibitors, are there any hints of these other factors interacting significantly with dose-dependent factors? And if so, or is that something that will need to be particularly considered for this class of drug in moving forward towards developing them further?
Hi, this is Nicole Gormley. I'll take a first stab at the question, and then I'll open it up to others on the team. So I think you bring up a lot of good questions, and these are things that we've definitely looked at to some degree. I will say we are somewhat limited, again, in that some of these, depending on what factors we're correlating the response with, some of these factors, we don't have the most robust information. But, this is something that we have seen and try to, further characterize. In particular, you noted that patients, we see a little bit of a strange, they're not strange, but just atypical factor or phenomena here, in that, a lot of the toxicities that we see tend to sometimes be worse with the PI3 kinase inhibitors in younger patients, or those that have received less, intensive therapies previously, or, newly diagnosed. And so we have a wide, data set here, but it is sometimes hard to pin down specifically what those factors are that are contributing the most or have the most impact with this class of products. I'll open it up to others that may want to respond from the FDA team.
Yeah, hi, this is Nicholas Richardson, FDA. Maybe just to reiterate what Dr. Gormley said. So you raised, an important concept and question with this particular class. And I guess just two comments. So one, we have, there is some data out there that supports that, patients with untreated or treatment naive disease and or those that have received less prior therapy, may be at greater risk for immune-mediated toxicities. And we have noted that trend in the PI3K inhibitors, and that trend also exists in other immunotherapy agents. And I think it's not clear, but maybe one example is the 0116 trial for idelalisib was in relapsed CLL, looking at idelalisib plus rituximab versus placebo and rituximab, and the safety profile and the safety outcomes for that particular trial, were different than the 0123 trial, which was evaluating idelalisib in a treatment-naive CLL population. And so we have noticed this trend in that regard, in which patients with treatment-naive disease or less prior therapies, do seem to be at increased risk and have numerically higher rates of immune-mediated toxicities. And then again, I think it also goes back to the specific isoforms and the selectivity of the agents, and do they impact one isoform versus others, and how that plays a role in the toxicity that we're seeing. And specifically with this class of agents, the impact on regulatory T cells really seems to be the primary driver of immune-mediated toxicities. So I think it's a really good question and comment. There is some data out there to support that, and it's something that should be noted and is important as we sort of look at the trials and development of these agents moving forward.
Okay, thank you very much.
Thank you. Move on to Dr. Chen. Andy, you have a question?
Thank you. This is Andy Chen, Oregon Health Science University. So with the withdrawal of duvelisib, idelalisib, and umbralisib, the only PI3K inhibitor left for follicular lymphoma is copanlisib. And from the Kronos 3 data that you presented, although the top-line hazard ratio for overall survival was slightly less than 1, you did point out that there's increased risk of deaths from adverse events. So is there thought on the FDA about having this sort of last option in this class to be withdrawn from market? Thank you.
So this is Nicole Gormley. I will, I can't, specifically comment per se on, what future regulatory actions will be. So I will note, though, that, several of the products do remain on the market. Some of these were indications that were drawn that were such that, the entire product was not removed. For example, idelalisib for the treatment of CLL, does remain on the market. And duvelisib is also currently on the market for CLL. And I don't know, I can have, I'm not sure if we have a slide. I'd have to think, sure if we have a slide that lists everything that's currently on the market. But so several of these products, do still remain, but these were just, indications that were removed. And as you mentioned, copanlisib also remains on the market. I think, where our perspective is, and I think, our concerns is that for anything that remains on the market, we have to have confidence that these products are safe and effective and that they don't do harm. In general, as was stated, earlier in our presentations, the onus is really on the sponsors to prove and provide evidence, substantial evidence that their products are safe and effective. And, that is the guiding principle, not how many are on the market, not what, is the product safe and effective? That's our regulatory standard. I hope that answers your question.
Thank you.
Jorge Garcia, Simon Cancer Center. I have a question for Dr. Gormley. So there's no doubt that we're interested in patients with prolonged natural diseases with prolonged natural history, such as the ones that we're reviewing today. Ideally, you want to have an earlier initial endpoint that can capture the overall outcome. And clearly, to me, at the very least, it's not clear that PFS is a valid surrogate endpoint for survival with very limited circumstances. I know in the past the agency has rejected time to treatment failure as a surrogate endpoint, but perhaps, and my question to you is, since we're talking about different class of agents with different mechanisms of action, I think that endpoints that can capture both dropouts due to toxicity, dose discontinuation, or dose reductions, and also because of tumor progression as events, may be ideal for this class of agents. So would the agency be open to look at different endpoints for this class of agents in particular?
So you brought up a lot of, really interesting and important points with the question about, what are the appropriate endpoints for, this disease space. So I'm going to take this in parts, and if that's okay. So the very first issue is that, in general, we are very amenable at the FDA to discussing with sponsors what the appropriate endpoints are for their trial design, for their specific product. We're also very open to engaging with the broader community and multiple stakeholders, industry, academia, et cetera, on development of new endpoints. And especially, early endpoints. That's generally, what's needed is development of earlier endpoints that can allow for, more expeditious drug development. But when we look at early endpoints and when we use those earlier endpoints, whether or not it's a surrogate or not, or just, an earlier intermediate clinical endpoint, we still always also need to look at overall survival. And so, and there's lots of ways that this can be done, but, trials ideally with an earlier endpoint is used should continue to be followed for later endpoints such as overall survival, such that we can fully assess them. And the true, clinical benefit from overall survival assessments. I'd like actually to have our statistical team, and I'll come back and make perhaps a few additional comments at the end, but I'd like our statistical team to comment a little bit as well about how we look at OS, especially also when it's not the primary endpoint. So I'd like to have Dr. Rodriguez speak.
Hi, thank you. My name is Lisa Rodriguez, and I'm the Deputy Division Director for Division of Biometrics 9 at FDA. I will give a brief overview of considerations for overall survival evaluations in general and for this PI3K class of drugs. Next slide. Overall survival is an important metric in supporting a benefit risk determination. So I will refer to OS
for overall survival in these slides. Here we will overview the regulatory viewpoint of this endpoint first. OS is typically defined as the time from randomization to death from any cause. Randomization tends to balance all factors, known or unknown. OS is a preferred efficacy and safety endpoint in oncology clinical trials. It is an objective measure of clinical benefit and incorporates the impact of toxicity. When pre-specified for hypothesis testing, the non-parametric log rank test has typically been used as a statistical test for evaluating significant differences in survival between treatments. OS is typically summarized via the hazard ratio and comparisons of median survival time. According to convention in oncology settings, hazard ratios are calculated such that values exceeding 1 indicate higher risk of death for the investigative treatment group. Confidence intervals for the hazard ratio are evaluated in the absence of or in addition to a statistical test. Other descriptions, such as the probability of surviving to set time points, can also be useful. A pre-specified ITT analysis is preferred for OS. However, we do typically conduct additional sensitivity analyses to evaluate the robustness of the estimates. Finally, OS is an important endpoint because it supports the overall benefit-risk determination for regulatory decisions. It is a safety endpoint as well as an efficacy endpoint. Next slide. Here I would like to outline some general issues associated with evaluating OS data and safety considerations that may be observed. A long natural history of certain diseases has motivated use of primary endpoints other than OS for efficacy claims due to the time needed to observe a sufficient number of events. Statistical analysis plans for trials using progression-free survival or overall response rate as primary endpoints have not always included event-driven pre-specified OS analyses. Because of this, hazard ratio interpretation may be challenging due to patients crossing over to subsequent treatments. There may be potential confounding due to subsequent therapies. There may be a low ratio of events to sample size, and OS is usually considered exploratory in such settings. OS is a safety consideration in general, and especially in the situation as we observe in the PI3K inhibitor class, where we can observe the following. A pattern of OS hazard ratios greater than 1, that is in more than one study. Prior information on risk for a product is informative, such as adverse events or risk of death, and there are label warnings for the PI3K inhibitor class. OS contributes to the totality of evidence and informs safety even in the absence of statistical testing. So, even with early OS data, the observed results, prior safety information, and observed toxicity profiles should adequately rule out harm and help support a conclusion that the products are safe. Next slide. These are some considerations we use to evaluate the OS data for the PI3K inhibitor class and may be useful to consider in general. First, it is useful to consider the available survival information based on the planned and available data. Even if none of the studies for this class specified a number of events for evaluation of survival data, and there were a low number of observed events as low as 3% of the planned sample size, leading to uncertainty in estimates. The estimated hazard ratios and confidence intervals provided descriptive information for the evaluation of potential safety signals. Point estimates for the hazard ratio exceed 1 across multiple studies. Wide confidence intervals do not adequately rule out potential harm. Death rates by treatment arm provide important summaries as well. As we saw from the main presentation, death rates were higher in investigative treatment arms for most of the studies. Next slide. In summary, regarding the evaluation of OS, the confidence intervals for the OS hazard ratio are wide with large upper bounds. However, we can observe that the large upper bounds indicate death hazards may be up to multiple times that in the control arm. There are higher death rates in the investigative treatment groups and higher OS hazard ratio estimates in several studies across the PI3K inhibitor class. While there are a low number of events and uncertainty in the estimates, when potentially harmful OS hazard ratios are observed in multiple studies in this class, the chance finding is questionable. Next slide. In summary, sponsors have an obligation to demonstrate their products are safe and effective. For the PI3K inhibitors, the observed overall survival estimates, especially considering prior information, observed toxicity profiles, and questionable dose selection, do not adequately rule out harm or support a conclusion that these products are safe. Thank you.
So this is Nicole Gormley again. Just to wrap up, I think, we are definitely, amenable and interested in exploring further and endpoints that could be used at earlier time points and that could expedite drug development. I think there's lots of potential candidates out there that could, be useful in this disease space. But even if those are used, we still really need to have an evaluation of OS because it's so critical to assessing, true clinical benefit risk. Thanks.
Thank you. I think Dr. Sung, you had your hand raised. You have a question?
Sorry, it was answered. Sorry, it was answered with my hand.
Right. Great, thank you. The committee will now turn its attention to address the task at hand, the careful consideration of the data before the committee, as well as the public comments. We will proceed with the questions to the committee and panel discussion and vote. I would like to remind public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. So this is question 1 for the committee, and the task at hand right now is for us as a group, after a pretty robust session of questions and answers and a great presentation by the FDA, for us to discuss the observed toxicity of the PI3 kinase inhibitors as a class and whether randomized data are warranted with an assessment of OS to support the evaluation of benefit risk in patients with hematologic malignancies. Are there any issues or questions about the wording of this question? If there are no questions or comments concerning the wording of the question, we will now open the question for discussion. Maybe I can start asking maybe an ignorant question as a drug developer myself. Maybe I can, if I can ask Dr. Conaway or Dr. Coffey to help me understand a bit of this. When you look at, Dr. Conaway, you mentioned earlier that there are multiple ways that you can do trial designs to address the questions that we all have and we sort of saw today in the presentations. I think one of my concerns is censoring when you are actually doing these clinical trials, especially single-arm studies. What is the true effect of when you have a higher dropout rate in the experimental arm, if you will, that either is due related to poor drug tolerability, and therefore patients need to actually come off trial before even you document progression, how that censoring or lack thereof affects PFS and ultimately, impacts outcomes survival-wise.
Yeah, and I'll address, my comments were really about the dose optimization phase of this, not the comparative phase, and I'll defer to others, Dr. Coffey or at the FDA for how they handle PFS and OS in the presence of censoring due to tolerability.
Hi. This is Nicole Gormley. I'll ask Dr. Gwai to comment.
Yeah, hi, this is Thomas Grimes, FDA.
In the face of censoring that's motivated by early dropouts due to toxicity, PFS could potentially be biased. In reviewing the studies that we get with PFS, we always do some sensitivity analysis to evaluate the amount of bias that could be caused by such informative censoring.
Thank you. Does anybody else in the panel have any comments, questions about what you saw with the toxicity data and how that can impact subsequent therapy and therefore maybe even outcome? Dr. Nieva?
Thank you, George Nieva, USC. I want to clarify that the question at hand really relates to other malignancies outside of chronic hematologic malignancies. That what we're really asking here is whether a PI3 kinase inhibitor, even if it's demonstrated to have a benefit in an area of unmet need, let's say they were found to be highly effective for glioblastoma or some other tumor for which there is a desperate need for better therapy, that option would be restricted and there would not be any accelerated approvals granted in that case. And really we're moving beyond the question of hematologic malignancy. So I guess my question is the issue of whether or not there is an unmet need moot? And is that really what you're asking? Thank you.
Hi, this is Nicole Gormley. Thanks. Our question here really is limited to, the use of PI3 kinase inhibitors in heme malignancies. There's obviously lessons that we could, from this discussion, that we will take back and think about and how that applies to other areas within drug development. But the question to the committee here is really, and the discussion is really just the PI3 kinase inhibitors and whether randomized data is needed randomized data with an assessment of OS is needed for patients with heme malignancies. I think, where we're coming from here is that, we have this body of experience with these products in these class in multiple, indolent lymphomas that have shown, significant toxicities and, concerns with dosing. And I think that has, implications for future exploration of PI3 kinase inhibitors within heme malignancies. And of course, again, yes, we will take back conversations here and apply them or, think about how they may apply to other, scenarios, other indolent diseases or PI3 kinase development in other spaces. But the question here today is based on the experience that we have, what should we be doing with PI3 kinase inhibitors in the future in heme malignancies? One other aspect that I want to point out is you mentioned in your question that, are we ruling out accelerated approval, for all future development, et cetera. And just one other important point to highlight is that accelerated approval does not have to equate with single-arm trials. So accelerated approval can be based on randomized trials still using early endpoints, and that's something that we have encouraged a paradigm for with multiple sponsors, that they consider an initial, for example, randomized trial that's powered for both early efficacy endpoints such as response rate and powered for later endpoints such as progression-free survival, such that one single trial is used and sponsors would come in with the randomized data for accelerated approval after having met the response rate endpoint, and then those patients we continued to be followed for overall survival or progression-free survival for regular approval once that data is available, but with single trial models. So the requirement or the question asking about randomized data really is separate from the question of accelerated approval or not. So just to highlight those two things. I think those are all the comments. I'll open it up to see if there's anyone else from the FDA that wants to comment. Okay, hearing none. Thank you.
Thank you. That was complete.
Thank you. Dr. Dhanarajasingham?
Thanks, Dr. Garcia. I just wanted to make kind of some summary comments, putting all of this together from my perspective, both as a lymphoma hematologist and a researcher focused in understanding toxicity and tolerability. And, my perspective here is that no one is arguing that there's not a clear efficacy signal or that development of this drug class should be halted and they shouldn't be available to our patients. The question is whether randomized data are warranted as a regulatory strategy here. And so, as a clinician, these aren't drugs that I'm reaching for initially, but they're ones that I would like to have available as options for my patients in later lines, usually after exhausting other available options. But for patients in later lines of therapy whose life expectancy is the most limited by their disease, the benefit-risk assessment is still very crucial, and it's still first do no harm for this precious population of patients. There's a concerning pattern of results here related to PFS benefits that lead to approvals and potential OS decrements that warrant additional scrutiny in the context of accompanying information about disproportionate toxicity and deaths in the PI3 kinase inhibitor treatment arms of several studies across the board. There's more than one reason why PFS and OS don't track, but the most concerning of them is treatment-related toxicity and deaths, and here the potential for harm can't be ruled out. It's interesting that efforts to gain clarity on these findings have not routinely panned out. The required post-marketing studies to affirm initial accelerated approvals haven't always been conducted, or the drugs are being pulled from consideration before the public and scientific community can get a clear understanding of why in scientifically rigorous peer-reviewed publications. So there's also very legitimate concerns that we've discussed about dose optimization. So a lot there, but I think although the voting question focuses on what type of trial is needed, I also, like Dr. Garcia, think that the type of endpoints we need to look at will be important. We all know that requiring OS endpoints in indolent lymphoid malignancies is impractical, and the intent is not to stifle progress and the speed to which therapies come to our patients, but sole dependence on PFS with these studies is problematic, and we really have to consider some composite outcomes that include PFS along with predefined safety and tolerability endpoints, which are informed by patient-reported data as well. And I feel that given the unique issues discussed today pertaining to this class of agents in further drug development, I would hope that the FDA might be able to require some of those elements, even if that's not standardly the case. So in summary, I do think that to understand the benefit-risk ratio in patients with hematologic malignancies, randomized data where possible is very important. So I'm very, very interested in hearing from my other colleagues on the panel. Thank you.
Excellent early summary, Dr. Dhanarajasingham. Fantastic. Thank you. Let's move up with Dr. Chang.
Thanks, Dr. Sear. I'm John Chang, industry rep. This is actually a clarification for Dr. Gormley regarding the question. So I appreciate the question regarding randomized data. My question is regarding assessment of overall survival as to how the FDA was thinking about assessment, particularly in situations where it's an indolent disease. And because the question is on, or the discussion is on the assessment of it and how an assessment is done, particularly if it's not a properly powered overall survival endpoint, which often is very large studies, and obviously intervening treatments can complicate that. So I'm interested in a less powered overall survival data set, how an assessment is potentially interpreted.
Yes.
And if I may just, as a secondary, is this because driven by the toxicity seen with this class, and would it be relevant to classes that are maybe have a more a safety profile that's distinct and maybe not with a less toxic profile?
Hi, this is Nicole Gormley. Thank you for that really good question. You know, we've had a lot of discussion about that here in the agency, and you know, we are, you know, with this meeting here talking about this class of products and their development in hematologic malignancies. But I think, you know, these discussions really have implications, as you stated, for many indolent diseases, you know, where you can't rely on overall survival as the primary endpoint. And you know, sometimes we may have early overall survival data. So I'd like to ask Dr. Rodriguez to speak briefly about, you know, how we're thinking about some of these overall survival assessments in going forward. Dr. Rodriguez?
Hi, thank you. So we do have some considerations for future studies. Next slide. Looking forward to future studies that can evaluate overall survival in randomized studies, we have some statistical points to consider. While FDA has demonstrated its commitment to timely approval of safe and effective cancer treatments through the use of earlier endpoints, survival is the paramount objective for interventions. A plan for evaluating OS should be pre-specified in the protocol when designing studies, even if not conducting hypothesis testing for efficacy. A pre-specified plan will be useful for a safety evaluation of OS in which potential harm to patients may be adequately ruled out based on a pre-specified data cut. Sponsors have an obligation to demonstrate their products are safe and effective. Approaches to early assessment and interpretation of OS may be useful, such as adapting trial monitoring approaches that may include utility analyses or Bayesian prediction. So these are our primary considerations at this point for future studies. I may also summarize slide 75 again, which covered what we looked at for the PI3K inhibitor class. So these were not pre-specified. These were exploratory analyses. Based on a low number of events, we did have uncertainty, but we did focus on the estimated hazard ratio and confidence interval to provide descriptive information. We also looked at the death rates by treatment arm to provide important summaries. So this was the basis of the overall survival and what we were thinking about in future studies. I hope that answered the question.
It does, and thank you for that. And is it based on also, is there different potential thoughts as to based on the toxicity safety profile of an agent or a class, or is this kind of universal?
I think these are general thoughts in terms of what we're looking at when we particularly have early OS data and this exploratory analysis where we did not have a pre-specified data cut, which would be a pre-specified number of events, and we were not conducting hypothesis testing.
Understood. Thank you very much.
Thank you both. Dr. Sung?
Anthony Sung, Duke University. Just taking a step back, I feel like this discussion is different from a lot of other ODAC meetings where, you know, we meet and we discuss a specific drug and the safety and efficacy of a specific drug. And while during this meeting, a lot of data was presented on a number of PI3 kinases, which I agree are problematic. I just, part of me struggles with, you know, how, with the question at hand and implications for the class as a whole and for future drug developments within this class. You know, what if a new PI3 kinase is developed that has phenomenal single-arm data? Would we still require a randomized trial in that setting? You know, I feel like usually when we meet on questions and we evaluate the data at hand, which I feel comfortable doing. But I feel like this question is asking us about the future, which is a little bit different. Thank you.
This is Nicole Gormley, and you're absolutely right. This is, a very different advisory committee meeting. And it is, future thinking and forward looking. And, I think, though, where we're coming from is that, we want to make sure that, our forward thinking advice that we give to sponsors is grounded in our experience. And, we want to make sure that we are learning the appropriate lessons, from this experience. I mean, in reality, this degree of safety findings that we're seeing and overall survival results across multiple products, across, multiple hematologic indications, and all showing this consistent finding of, concerning overall survival patterns, albeit early, is really unprecedented. And so while we definitely want to expedite drug development and make sure that there are, new therapies available to patients as soon as possible, it's imperative in our view that we ensure that those products are safe and effective. And so there's many ways that, that drug development can be expedited, and it doesn't all require a single-arm trial. I mentioned how, randomized trials can also be used for accelerated approval, and we have lots of other mechanisms to expedite drug development and work with sponsors as well to expedite development, and then also processes and programs to expedite our review for really effective therapies. But I think the issue that we're seeing here is that, all of these products have activity, but they also have a very concerning safety profile that really has only been able to be fully characterized with the randomized data. And so our question is, given, and again, this unprecedented body of data that we have thus far, going forward, should we require randomized data?
If I may follow up?
Sure, go ahead.
I guess my question is, like I said, I agree with you on all the data of the drugs that have been presented to date, and I share your concerns, but my question is, we can't predict the future. And what if the future, another drug in this class comes along that appears to have phenomenal safety data as well as, a strong suggestion of efficacy even in phase 1 studies? And can that go on to just a single-arm phase 2 study? I feel like it's hard to make. That's why I find it hard to struggle with this question, because it involves the future, and we don't know what's going to happen down the line.
No, we definitely don't know what the future is. I will say, though, that, none of us have a crystal ball, right? But, we are also not automatons. If the cure for cancer, is developed tomorrow, I think we can find ways to review this expeditiously, to study it expeditiously, et cetera. None of us has a crystal ball. But I do think that, it's really important that we don't make the same mistakes from yesterday, that we learn from our experience.
This is Dr. Pastor. Let me just jump in here. I think you're kind of reading into this question in a little more detail than we intended. Of course, we would demonstrate the appropriate degree of flexibility depending upon the safety findings as well as the efficacy findings, that's for sure. But if something similar came along where we saw, even in the early studies, a significant toxicity here, then this should raise concern for us with regards to potential impacts in overall survival. So we're not asking you, as we said, we realize that you do not have degrees in fortune telling here, so to speak, but given the fact of what we're seeing here, and here again, this is an unprecedented finding that we saw in oncology here. If we saw something similar as we move forward, should we do randomized studies? Granted, if we saw something that had phenomenal response rates and that were very non-toxic, then that's a different story here, and we always would demonstrate the appropriate degree of flexibility in this.
And so that's something that I agree with. It's just not how the question is stated.
Again,
duly noted, okay?
Yeah.
Thank you all. Next, Dr. Coffey.
Yeah, I just have I guess a clarifying question where a couple of comments, I'm having trouble reconciling a couple of the comments that have come up in the discussion where it's been mentioned multiple times that like in randomized trials, you could still do accelerated approval. But the emphasis on overall survival, I mean, it was referenced, several times in the discussion, the wide confidence intervals that you have, right, even in the existing data. And with the approaches that were mentioned with like a futility analysis or some type of Bayesian prediction to stop, you're still going to need a decent number of events to do that. So it seems like if that's the direction that future trials are going, that is almost by definition pushing you away from accelerated approval because you're going to need longer studies before you would do that. So I guess I'm trying to reconcile those two statements in my head and just wonder if anyone might want to comment on that.
If I may interject, we're close to time and we have still a voting question, so I would ask the group, whoever is left or answering questions or comments, to keep them brief and succinct, please.
Yes, this is Nicole. I'm sorry, could you clarify your question again? Is it the distinction is just about the randomized data versus, could you just clarify your question again?
My question was more if the futility, the plans for future trials with overall survival and randomized, if you're using like futility rules or Bayesian prediction to try to stop them, to have stopping rules, that's going to take a number of events, right? With the wide confidence intervals that you report in the studies that have been done, it would be hard to have reasonable stopping rules in those, which seems to push it away from the option of accelerated approval that's been mentioned numerous times is still on the table. So it almost seems by definition, if you go in that approach, the accelerated approval is going to be a much harder pathway just because of the numbers aren't going to be there. So I'm trying to reconcile how could you do
Not
that type of...
necessarily. Obviously, you would take a look at a accelerated approval, for example, on a response rate, which would require fewer numbers of patients than an overall survival analysis or even a time to progression analysis. This is Dr. Pazder. But you actually have the trial ongoing, okay? So you could actually see these effects later on, okay? So this is, rather than doing these trials sequentially, a randomized trial versus this continuation of a randomized trial in one trial, one actually has the trial ongoing. So we're not saying that we would hold up an accelerated approval necessarily for a survival analysis, but they would be forthcoming relatively rapidly, one would hope. Certainly not a year or 2 years later, that's for sure. Or many years later.
Thanks. That clarifies.
So here again, it's the true success of accelerated approval is basically to try to shorten the period of time between the designation of accelerated approval and basically the confirmation of clinical benefit or lack of confirmation of clinical benefit. So this single study approach where we have an accelerated approval on a response rate and earlier clinical endpoint is one that we're really advocating. Then you do have the trial ongoing, so there's none of this issue of, well, it's going to take us several years even to get a randomized trial ongoing in a specific disease. And then you have drugs out there that potentially are harming people with a long period of time on the market. That's what our interest is in here. Clear?
Thank you, Dr. Pazder. We have one final comment. Dr. Deal? Dr. Deal, you may be muted still.
Can you define the word warranted in question 1? For example, does it mean the defining factor is overall survival, or it will be considered, or it should be pre-planned? What does the word mean, warranted?
Yeah, this is Nicole Gormley. The warranted was, again, referring to the randomized data. I mean, it's randomized data are warranted, and it should include an assessment of overall survival. We are not, suggesting that overall survival be the primary endpoint. Again, as stated earlier, what we're really after is, or asking here is, should we have randomized data for initial approval? And again, accelerated approval can still be used with a response rate, but with a randomized trial, and those trials, followed for overall survival. And then, again, as mentioned earlier, initial looks, early looks, interim futility analyses for overall survival as well throughout the trial. But it's just, the amount of information available from randomized data is so much more robust, including, patient-informed outcomes, a better assessment of safety and attribution of the toxicity observed, tolerability, et cetera, across the board from randomized data as compared to single-arm trials. And so given the experience that we've seen thus far, none of these trials evaluated overall survival as the primary endpoint, and that's not what we're suggesting. The question is just, or the discussion point is, should randomized data be required for initial approval and with some looks or assessments of overall survival throughout with that trial?
Thank you.
Thank you. So just before we move on to question 2, which is a voting question, let me just briefly summarize what the panel has reviewed. It does appear that efficacy signal does exist, that we think that these agents do provide efficacy. The biggest issue, obviously, is tolerability, based upon the data that we have seen throughout all these class of agents. The benefit and risk assessment remains critical for drug development in this context. Certainly for patients with a prolonged natural history, and certainly in the context of second and third line therapy as well. Where also some comments about the patterns for the hazard ratios for survival are concerning, and including of that, the frequent withdrawal of agents that appear to be concerning and legitimate as well. And finally, perhaps the need for us as a group and drug developers in the country to sort of innovate with new endpoints and certainly include PROs in all these clinical trials. And perhaps for me, there is no doubt that patients want timely access to new cancer therapies, but certainly they have to expect that us as investigators identify those therapies that offer real benefits in their lifetime. So we move on to the next question. So this is a voting question. Given the observed toxicities with this class, previous randomized trials with a potential detriment in OS, and a narrow range between effective and toxic doses, should future approvals of PI3 kinase inhibitors be supported by randomized data? Dr. Shixia Shen will provide instructions for the voting.
Hi, this is Shixia Chen. Question 2 is a voting question. Voting members will use the Adobe Connect platform to submit their votes for this meeting. After the chairperson has read the voting question into the record, and all questions and discussion regarding the wording of the vote question are complete, the chairperson will announce that voting will begin. If you are a voting member, you will be moved to a breakout room. A new display will appear where you can submit your vote. There will be no discussion in the breakout room. You should select the radio button that is round circular button in the window. That corresponds to your vote, yes, no, or abstain. You should not leave the no vote choice selected. Please know that you do not need to submit or send your vote. Again, you need only to select the radio button that corresponds to your vote. You will have the opportunity to change your vote until the vote is announced as closed. Once all voting members have selected their vote, I will announce that the vote is closed. Next, the vote results will be displayed on the screen. I will read the vote results from the screen into the record. Thereafter, the chairperson will go down the roster and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. Are there any questions about the voting process before we begin?
I'm going to read the question again. Given the observed toxicities with this class, previous randomized trials with a potential detriment in overall survival, and a narrow range between effective and toxic doses should future approvals of PI3 kinase inhibitors be supported by randomized data. Are there any questions about the wording of the question? If there are no questions or comments concerning the wording of the question, we will now begin the voting on question 2.
Sorry, this is Anthony Sung from Duke. I did raise my hand. So, Dr. Pazdur, should we vote on the question as it's written or on the sense of the question as you had previously articulated?
Well, the issue here, I think they're not inconsistent here. We're just asking, should randomized studies be done here? We're not asking for overall survival to be the primary endpoint of the trial, but a randomized trial does allow us to at least do a descriptive analysis of that endpoint that cannot be obtained from a single-arm trial. So this is, I view this question as totally consistent with my previous comments.
I guess in my mind, there's the inconsistency is, previously you had said if there's phase 1 data that, raises some concerns, then we should do a randomized study, which I absolutely agree with.
Here again, we would have the flexibility here. This is given this, I think it's well worded here, given the observed toxicities, okay. This was a toxic regimen. We had, potential detriments in overall survival here. So in general, would people support a randomized study? Obviously, there are exceptions to anything, and we would demonstrate the appropriate flexibility depending on what we saw in these earlier studies here. But given this class of drugs here, if you had to do this development plan over, I think most people would agree, and not to lead the committee here, but most people would agree that there should have been randomized studies here, obviously, done earlier.
And I absolutely agree with these drugs that have been presented, but if a future drug does not show toxicity in phase 1 studies,
Good evening, everybody. This is a DFO, Shija. Just a friendly reminder, please read a vote as the question it is. Vote as what it is, and we can go ahead and moving on. Thank you so much. And I'll pass to Dr. Garcia.
There is opportunity after you vote, right, for you to state and comment as to why you voted. So please save those comments for after your vote, if you will. Just vote as the question reads.
A quick question. Are we voting on the overall randomized trials of 2 different doses?
The question to me is clear. So I would suggest for you to vote based upon what the question states, and then you can actually think as to why you voted the way that you voted after, and make comments regarding that after you vote. Dr. Shen, you want to take us to?
Great, thank you. We will now move voting members to the voting breakout room to vote only. There will be no discussion in the voting breakout room.
Sub-conference rooms are open.
Voting has closed and is now complete. Once the vote results display, I will read the vote results into the record. The vote results are displayed. I will read the vote totals into the record. There are a total of 16 yeses, 0 no, and 1 abstention. The chairperson will go down the list and each voting member will state your name and their vote into the record. You can also state the reason why you voted as you did if you want to.
Thank you. We will now go down the list and have everyone who voted to state their name and vote into the record. You may also provide justification for your vote if you wish to. We'll start with Dr. Chen. Andy?
Andy Chen, I voted yes.
Thank you. Dr. Sung?
Anthony Sung. I abstained for partly the reasons that we had already discussed, but to summarize here, I agree that the drugs that have been evaluated in this class and discussed today are highly problematic, and how those evaluations were done has its flaws, and randomized studies should have been done in that context. However, I still feel uncomfortable labeling an entire class and requiring further future drugs in that class to be supported by randomized data. I think if the phase 1 data is concerning, then absolutely a randomized study should be needed. If the phase 1 data is not concerning, then I don't know if a randomized study should be needed in that case. Thank you.
Thank you. Dr. Coffey?
Yes, Chris Coffey, yes.
Dr. Liu?
This is Chris Liu. I voted yes. I think, when you look at the significant concern that overall survival endpoints for indolent cancers, I mean, this can be costly, extremely time-consuming, and I think that the utilization of PFS benefit as an endpoint for regulatory approval is potentially more reasonable with therapies with limited toxicity. In this case, I think it's likely not reasonable in a situation where therapies have significant toxicities, and also agents with significant toxicities may lead to the potential confounders to progression-free survival, as has been brought up during the course of this call.
And also with the available data, you would at least hope to see at least a trend towards overall survival, even with subsequent lines of therapy confounding that. But with this class of agents, in some trials, the reverse actually appears to be true, further highlighting the concerns that are raised today. And the bottom line is, if we aren't improving length of life with any therapy, but exposing patients to toxicity and therefore decreasing their quality of life, are we truly helping our patients? And I don't believe so. This concludes my comments. Thank you.
Thank you. Mr. Mitchell?
Yes, I'm David Mitchell. I voted yes. I think we need randomized trials to ensure that the products we're addressing today are safe and effective and don't do harm.
Thank you. Dr. Singham?
This is Geeta Thanarajasingham. I voted yes, and I don't have anything to add to my prior summary comments.
Thank you. Dr. Al?
I'm Jessie Al. I voted yes.
Thank you. Jorge Garcia, I voted yes, multiple points. I really believe, and it's perplexing to me, the lack of appropriate dose escalation studies for these agents, especially when they're using combination with other existing regimens for those diseases. Certainly the AE profile and the reduction in dose and drug discontinuation as presented by the group is quite perplexing to me as well, and quite toxic in my mind. Also, the trend to survival detriment, it is something, again, that is perplexing to me. And the reality of it is, as I think clinically, even though I'm not a hematologist myself, I could probably find quite difficult to tell a patient that I have an agent that could reduce your tumor volume, possibly delay your progression, but at the price of significant toxicities. And by the way, I can also impact your mortality in a detrimental manner. Dr. Nieva?
George Nieva from USC. I voted yes. Randomized data are always ideal to show efficacy and safety. Single-arm data is valuable for approval of novel agents and in areas of unmet need. The question is worded as specific to PI3 kinase inhibitors for chronic hematologic malignancies, where we now have lots of agents on the market, so there is no unmet need, where approval based on a single-arm study would have been sufficient. The current safety data justify raising the bar for new agents in the class to show that they are not causing long-term harm. I am concerned, however, that the selection of study endpoints may have impacted toxicity. In indolent lymphoproliferative disorders in particular, it should be noted that the PFS may have biased drug design for longer-term drug administration rather than fixed-dose administration, and this long-term administration for these drugs may have been detrimental to patients. Response rate may have been a preferable endpoint in these disorders, and there should be attention to endpoints that reflect clinical benefit with shorter-term administration. Thank you.
Thank you. Dr. Dunleavy?
Hi, I'm Kieran Dunleavy. I voted yes. I have no further comments to add to my previous comments.
Thank you. Dr. Diel?
Blue deal. The speakers made a compelling case, and the solution that they proposed, a randomized trial, go a long way towards solving the problem. Thank you.
Thank you. Dr. Conaway?
Mark Conaway. I voted yes. I think the results presented provide ample evidence that randomized trials should be part of the approval process for PI3K inhibitors. I think they also highlight the need for improvements in the design, conduct, and reporting of the dose exploration trials leading up to the randomized trial.
Thank you. Dr. Cristofanilli?
Yeah, Massimo Cristofanilli, I voted yes. I think it's very clear from the studies that there is a class effect toxicity that we need to keep in mind for the future with regard to dose-finding studies. And the randomized study is the only way to address acute and chronic toxicity to see if these drugs have a future in hematological malignancy.
Thank you. Ms. Nadim Baker?
This is Michelle Nadine Baker. I vote yes. for the reasons already stated by Dr. Liu, and although I don't want to stand in the way of progress with drugs, the evidence presented is very compelling that this class of drugs needs to be supported by randomized data. Thank you.
Thank you. Dr. Advani?
I voted yes for the reasons already stated by my colleagues on this call, and I was reassured by Dr. Pazner's comment about having flexibility in case the next agent in this class of drugs comes along, which has amazing activity, that there might be some flexibility. Thank you.
Thank you. Dr. Madan? Dr. Madan?
Yes, this is Robbie Madden. I voted yes. I think the patients, I think the historical experience here really begs for randomized data. And I think our patients have an expectation not just to live longer, but to live as long as they can and maintain a quality of life. And randomized data will provide confidence for physicians and patients alike to provide that.
Thank you. Dr. Kraft?
This is Walter Kraft. My vote is yes. The well-established power and benefits of randomization and evidence generation strongly outweigh disadvantages of this approach in the case of PI3K inhibitors and in the current therapeutic landscape. Thank you.
Thank you all. So we have, again, just to summarize, we have 16 yes and 1 abstain. Pretty much everybody who voted yes, just talking about the standard for clinical trial design to be randomized trials, the concerns of survival detriment, the benefit risk ratio in that patient population with long natural history, and the importance of quality of life as you prolong life for these patients. Certainly, PFS, at least for most of us who voted yes, didn't appear to be a reasonable endpoint for this class of agents. For the person who abstained, the concerns were simple and related to labeling a class of agents that in the future may pan out to be effective and safe for most patients. Before we adjourn, are there any last comments from the FDA?
This is Nicole Gormley. Thank you all for your comments. They're very insightful, and thank you for your time.
Thank you. I would like to thank the FDA for an excellent presentation, the committee members for an active session of questions and a robust discussion, despite of some questions about the questions at hand, and certainly the FDA and ODAC staff for making this meeting possible. We will now adjourn the meeting. Thank you all, and stay safe.
The meeting is now over.